Une petite annonce pour les gens, les anciens étudiants du master Bioinfo de Rouen, ainsi que toutes les personnes qui ont participé au master. À 10h20, on se retrouve sur la terrasse pour prendre une photo souvenir. Merci, bonne journée.
So good morning, everyone. It's time to start our second day of JOBIM with exciting talks. But we will start right now with Laura Cantini, who is our keynote speaker for this morning. So Laura was originally trained as a mathematician, not a bioinformatician, but she did a PhD in Italy. She then joined Emmanuel Barrios' group at the Institut Curie for her postdoc. She spent also some time at MIT. She was then recruited as a CNRS researcher in 2018 at the ENS in Paris. She has held a Prairie Chair for Artificial Intelligence since 2021, and she established her own group at the Institut Pasteur in 2023. So over the past few years, Laura, I think, has really become one of the leading researchers of her generation in computational genomics. So her group really develops computational machine learning approaches to integrate single-cell multi-omics data, reconstruct gene regulatory networks, and it's really exciting to have give you a talk today about this. So her achievements have been really recognized by numerous distinctions, including the L'Oréal UNESCO Fellowship for Women in Science, an ERC Studying Grant, and the CNRS Bronze Medal. So I'm very happy to have you here, Laura, and the floor is yours.
So thank you for the kind introduction, Anais. And thank you for the invitation also to give this keynote talk. Let's start from the overall motivation for the work of my team. One question that is deeply fascinating me and motivating the work of my team is to try to understand the identity and destiny of cells. And what I mean with this is that during development, we go from one egg cell to trillions of different cells that are extremely heterogeneous, phenotypically heterogeneous, and this allows them to fulfill different roles in the human body. So we can see cells as evolving in time along trajectories of continuous phenotypic transitions. And these trajectories will have some branching points to allow them to specialize into different directions. And we have still little knowledge to be able to reconstruct these trajectories, to be able to understand the full heterogeneity of these cells. So we are not able to distinguish what are the different cell states that appear during these trajectories. We are not able to reconstruct the path of the cells during these trajectories, and we don't fully understand the networks that can determine the decision-making of a cell along this trajectory, and in particular also branching points in the trajectories. And this is crucial for different biological processes, going from development to also disease, because it can help us to intercept early the onset of diseases and thus be able to better treat them, have better biomarkers to identify them earlier during the process. And why addressing this problem is well-timed? Because we have all the input data needed in order to address the problem. Thanks to single-cell technologies, we have large-scale measurements at single-cell resolution. And these include different omics data, so different types of molecular information that is complementary. We have many spatially and temporally resolved data, spatial proteomics, spatial transcriptomics. And each data set, however, is a huge data set with tens of thousands of variables, millions of cells that we have to analyze all at the same time. And so the work of my team and the fundamental knowledge gap that we have right now in the field is to try to define multimodal learning methods that are able to jointly integrate all these heterogeneous information in order to better characterize cell identities, cell trajectories, and the gene network that are determining these cell trajectories. And why new methods are needed for this? Because first of all, the single-cell data that we are dealing with are characterized by computational complexity coupled with large size. As I was saying, we are dealing with tens of thousands of features. The data are dramatically sparse and heterogeneous. Single-cell data present dropouts, which are zeros that we don't know whether they're real zeros or limitations of the sequencing machine. So we have 80–90% of the values of these measurements that are zero. and extremely heterogeneous. We are measuring different features. The data have completely different distributions, so it's difficult to understand how to combine these different distributions together. And then we are in an highly unsupervised setting. While, for example, when we do image analysis, frequently in the context of pathology, when we analyze H&D images, we frequently have pathologies that have annotated the images, and so we can use this to train supervised methods. In this context, if you want to identify cell types, cell states, we have little knowledge on all the markers that can identify different cell states. And so we cannot deal with supervised methods. And the work of my team has been focused on 4 main directions to address these problems. The first one is to integrate different omics data in order to reconstruct the different cell states and cell types that are present into a cell. And we will see some of these methods along my talk. Then the second direction is to reconstruct the regulatory mechanism that can explain the difference in between one group of cells and another one. Then we want to go into the dynamics and reconstruct cellular evolution through different single-cell measurements. And here we were using, in particular, spatial transcriptomics data, so also taking into account the spatial component. And finally, we went in the direction of building foundation models for single-cell data and trying to automatize this process after learning information from a lot of different data types. I will start from the first direction. And here, the first step in order to define cell types is to have a good metrics that can tell us whether cells are similar to each other. And for this, in the PhD work of Gertian, I'm not great in pointing, we have been testing the role of optimal transport for measuring similarities in between cells. The idea here is that we are, let's say, modeling cells as probability distributions, and then we are computing their distance as the minimal effort that you need to morph the gene expression distribution of one cell, for example, cell A, into the gene expression distribution of cell B. Biologically, this means that cells that belong to the same cell type activate more or less the same biological processes, so it should be sufficient to move a few bin of expression from cell A to cell B in order to make them similar. So we should detect that they are close to each other, their profiles are close to each other. And then we went to test it, and we tested it on simulated data and on real data. Here I'm showing you the results on real data set. We have single-cell RNA-seq data, single-cell attack, and single-cell methylation. All the data sets have labels. So in the case of the first data, for colorectal cancer, tumor and normal tissue, it's FACS-sorted cells. For cell lines, for attack, it's cell lines. So we have a clear definition of the groups that are present in the data. For methylation, it is defined by expert biologists that are looking at markers, expression, so it's the less well-defined case. These are the results in terms of adjusted rand index that we get after clustering this data with the standard method, and the standard method that we considered is SEORA, which is based on computing Euclidean distance in a PCA space in between the cells and then building a Kahneman graph and clustering this graph. And if we just substitute Euclidean distance in this method with optimal transport, that's the improvement that we can observe. So this really convinced us about the power of these distance metrics in order to evaluate similarity in between cells, and it was particularly interesting to see the performances that we observe in colorectal cancer normal, where there were rare populations that were really difficult to detect with the Euclidean distance approach, while we were greatly identifying them using optimal transport. Then the following step was to think about using these metrics in order to integrate different single-cell omics data. And we have been working in 2 major directions. The first one is to deal with paired data, which is a bit a simpler setting, because we have different omics data that will present different features, peaks, genes, but the same cells. And here we are with technologies like 10x Multiome or CytSeq. And then the second direction is instead to deal with unpaired data. So here we have different features in the data and also different cells. And the idea is to take advantage of all the independent sequencing of different omics data that has been done for years and that we want to take advantage of in order to better understand the heterogeneity that exists in between cells. Okay. So the idea is that we still want to use the complementarity between the different data types, but we are dealing with different cells. And how do we do it? So that's the PhD work of Jour, and we develop SC Confluence for this. Basically, the method is based, first of all, on the idea of reducing the dimensionality of the data. So here we're using Gaussian encoders in order to reduce the dimensionality of our data. You can think to X1 as single-cell RNA and X2 as single-cell attack. You want to reduce the dimensionality and reconstruct the data set that is as close as possible at the original one. But this is not sufficient alone, because if you just do this, you are not learning anything about how the cells of the two data sets are related to each other. And so to learn how they are related to each other, what we did is to use inverse optimal transport. So we have seen before the measure of the similarity in between the cells based on optimal transport. Here, the challenge that I was saying before is that the cells are represented by different features, so they're not comparable as they are. And so what we do, first of all, is to take a noisy translation of one data set into the form of the other. If you are integrating single-cell RNA and single-cell attack, this can be gene activity, which is a noisy representation of gene expression that you can build from attack data. This allows us to get a distance in between the cells in gene space, and then we can project this distance in the latent space, trying to identify the cost in the latent space that would give rise to a distance which is as close as possible to the one that we found in gene space. And this allows us to mix the cells in the latent space in a way that is informed by the known association in between peaks and genes. And then we went to benchmark this approach. I'm going to show you just a benchmarking in one of the data sets. What we see overall is that the methods take advantage of cases where there is a big difference in the terms of number of features of the data that you are integrating. So in this case, we are using site-seq data. These are paired data, but we are hiding to the algorithm the information that the cells are the same. And we are just using these for evaluation of the performances of the method. One dataset has only 23 features, and the other one has 10,000 genes that are expressed. And so there is a big difference in terms of features in between the two datasets. And other approaches would have just converted the features to the same entity. So they would have been working with only 23 features in the two datasets. What we do instead allows us to use for the autoencoder all the features of the dataset and just use the 23 in common to learn the alignment in between the cells in the latent space. And what we can see based on purity, which is telling us to which extent we are putting close to each other in the latent space cells from the same cell type, transfer accuracy, which is using the pairing information to test whether we are putting close to each other the corresponding cells of the data set, and mixing, which is evaluating the entropy in between the two data modalities. What we can see is that overall we have better performances than the alternative methods that are quite state-of-the-art, as Seurat, V3, Liger, Multimap, Glue, and so on. And here, just to give you an idea of the results of the integration, you have you have the latent space representation with your UMAP, with the annotation of the different cell types, and also the mixing in between the data modalities, and you can see that there is a good mixing in between the yellow and the blue cells. Then we want to try to challenge the method on some more interesting applications, and I'm going to show you just one of them here, which is based on single-cell RNA and SNFish integration. The idea is that single-cell RNA is a destructive technology, so we lose the tissue information, but it allows to do high-throughput sequencing, and so to measure a lot of genes in a lot of cells. SNFish, it's imaging-based, so we keep the tissue context of the gene expression, but given that it's based on fluorescence, we can measure way less genes, hundreds, 1,000 maximum. So what we did here is to use SC-Confluence to integrate the SNFish and the single-cell RNA data. And here you have the results of the embedding that we get with all the different cell types annotation. But then we could do something nice, which is to try to predict how they would look in space, genes that were only measured in the single-cell RNA data. So first, in order to benchmark the quality of the results, we have been hiding some genes from the SNFish experiment, trying, based on the integration, to predict the spatial profile, and then comparing the real spatial profile that has been hidden to the method with the prediction. And these are the results that we get. It's an example of 3 genes. On the top, you have the ground truth prediction, or the ground truth measurement, and on the bottom, you have the imputation, so the prediction done by the method. And it's nice to see that overall, the patterning of gene expression in the tissue is well reproduced by the method, with some exceptions. For example, the second gene has an area, here, where the expression goes down, and in the imputation, we are not able to detect this. So we can detect the major patterning of gene expression in space with some limitation. And then we could even do something more and take all the genes that were not measured in the SNFish experiment and try to see how they would look like in their expression in space. And we could identify genes that were having special patterns that are similar to those of the markers that were selected in the SNFish experiment. And that could be possibly interesting to investigate for future analysis. So now what I have shown until now is based either on using paired data, either on using unpaired data. The issue is that in real biological application, when you want to deal with rare population or transient populations, the paired data integration is not perfect, because given that it presents a lot of dropouts, it's really tough to get rare populations from paired single-cell sequencing. At the same time, unpaired integration allows you to have to deal with higher depth of sequencing data, but you have to use some knowledge of the interaction in between the features in order to do the integration, and also this is not perfect. So the next direction that corresponds to a new preprint that came out from the team and is still the results of the work of PhD work of Jour is the development of Champollion, which is combining unpaired integration with the use of few pair cells. So you can have even 10, 100 cells that are computed, that are profiled with 10x multiome, and this information of pairing can help to improve the integration of the unpaired data, and we think that this is the best solution in order to get rare populations. Then the second direction is cell foundation models. And for this, we're among the first lab proposing a foundation model for single-cell RNA data. That's the PhD work of Jeremy that recently finished his PhD. And we developed a tool which is called Sprint. So the idea here is to develop a foundation model which is based on a bidirectional transformer. What is taken in input are expression data from your genes, the location of the gene in the genome, so that it can understand, it can capture the fact that genes that are located close to each other in the genome might influence each other in expression, and the SM2 embedding, the protein embedding of the gene. The use of the protein embedding is just a substitution to the gene symbol, and the idea is that given that gene symbol change in between human and mouse and in between species, the use of the protein embedding allows us to generalize more easily to other species. And differently from other foundation models for single-cell RNA, like CGPT or SC Foundation, what we do is that we don't use masking for training the model, because we think that masking makes a lot of sense in languages where the order of the words in a sentence has a lot of meaning, but makes less sense in biology, ranking the genes based on their expression and then masking some of them. So we wanted to think about training tasks that were making more sense for biological applications, and we ended up with a combination of 3 tasks, and we are summing up the losses of the 3 of them. The first one is denoising as upsampling. So the idea here is that we want the method to understand that different technologies will give different data distributions and that there are dropouts in the data, so you might see some zeros that are not real zeros. So we are, let's say, downsampling the expression data of the cell and asking the method to reconstruct the full expression profile. Then we have bottleneck learning, which is based on the idea of compressing the information that is present in the cell expression data to lower dimension, and then being able to reconstruct the full cell expression profile as close as possible to the original one. And finally, classification. So the idea here is that we are dealing with data, we are using cell-by-gene data, from the Chang-Zuckerberg initiative, where we have 50 million cells profiled from different tissues, different disease contexts, and so on, with all the labels associated to them. And so we have really rich information that we can use to train our model in classifying these cells and then checking whether it's correctly detecting the right labels. And what are the outputs that we can get with Sprint? Obviously, we can get label prediction, given that it's trained on classification. We can get denoising, and this is also just a consequence of the training task. But then we can also reproduce deconvolved embedding, and so batch correct the data and reproduce embedding that capture the different cell types present into the data. And the most interesting part that we really care about is that we want to get gene networks. So the idea here is that if the model, with all the task of the clustering, is learning to classify cells, to compress them and so get the most information that is present into them, it should have understood what are the genes that it has to look to in order to make this prediction. And we think that this reflects, in fact, the gene network that is regulating the cell. And so we can get this gene network in 2 ways. The first one is to average the attention matrices. And the second, if you have few interactions that you know that they are true, then you can apply a classifier on top of the attention matrices, and this will help you to select a few links. And this is just to give you an example of the type of output. So you can get your embedding with all the different cell types and the annotation, and then gene networks that are associated to each group of cells. So also differently from methods like CNIC and so on that will give you a general network for all the cells, here you are getting cell type specific networks. We benchmark the method extensively, a lot of different tasks. For a matter of time here, I will show you just the results on gene network inference and just on one benchmarking. This is perturbation data, genome-wide perturbation data from a cell line, K562. And we have two metrics of evaluation, EPR and AUPRC. AUPRC will evaluate the full ranking of the links inferred by the method, while EPR will just look at the quality of the top links. On the left, you have methods like GYPSAM and Gini3, which are applied to the dataset itself in order to infer the gene network. Then we have a series of foundation models, GeneFormer, SCGPT, and our model Sprint, which are used with zero-shot prediction. So they have not seen the data themselves, they have been trained on other data, and then they are predicting the network in this dataset. So what we can see, first of all, just looking at the standard Sprint, is that it performs better than alternative foundation model in this task, but is still lower than Gini3, which has been designed originally for gene network inference and that has been run on the data themselves. Then what we try to do is to use the classifier. As I was saying, if you know few links, you can train a classifier on top of it. And so we thought, given that we have some prior knowledge, we have OmniPath that is giving us a lot of transcription factor gene interactions, let's train a classifier on OmniPath links. And what we see and what it was interesting to see is that in this case, the performances drop, indicating that the intersection in between the perturbation experiment and the OmniPath interaction is really small. And so training on this database, it doesn't help to improve performances. And then finally, we just try to train the classifier on few interactions that were present in the perturbation experiments. Obviously, we took out this interaction from the evaluation. And what we can see is that in this case, the performances go higher and become comparable with those of Gini3. So these were encouraging results, but we're still far from inferring good networks with foundation models. And just the next direction for this is that we have been working on a new version of Sprint, which is called Sprint 2, which is currently under revision. And there what we try to do is mainly to do an ablation study, given that a lot of foundation models now exist for single-cell data, and all of them are making different choices in terms of architectures, in terms of the data used for training, in terms of how the networks are inferred, and so on. What we do is an ablation study to test the added value of each component of these foundation models to then get the best model that can put together all the best components. In addition, in this work, we also increase the set of the training data. And so we don't just use the cell-by-gene data with mouse and human, but we use data from a lot of different species and also SARO data, which contain perturbation experiment. And we have some architectural novelty with expressor architecture. Then the second direction is more focused on gene regulatory network inference and combining different omics data in order to infer these networks. And in this direction, the main state of the art that was existing when we started working on this topic for inferring network from single-cell RNA and single-cell attack was based on regression models. Regression models applied jointly to single-cell RNA and single-cell attack data. And you have here the main methods that have been nicely reviewed in the work of Pau from the lab of Julio Saez Rodriguez. What we did was to try to use a different approach, to use multi-layer network to keep the different omics data separated, the network coming from different omics separated, and to see where this could allow us to get more rich information in the data. So the idea of multi-layer network is to represent interactions in between different elements and then their correspondences in between them. You can imagine that a classical application is in social science, when you want to study interactions in between people, and you are taking into account different channels of interaction, Facebook friendship, phone calls, and so on. So here our networks are genes for the single-cell RNA layer and correlation of expression in between them, co-accessibility in between attack peaks, and cooperation in between transcription factors, which is an intersection in between protein-protein interaction and chip-seq co-accessibility. And then we can define correspondences in between the nodes of the different networks. For transcription factor, we can connect them to the peaks based on motif, and we can connect the peaks to the genes based on the proximity of the peak to the starting site of the gene. And then what's the main advantage of this is that, as I was saying, you keep the information separated, so you can take into account cooperation in between biological macromolecules that is lost once you project all these in a single network. It can easily include other omics, and it doesn't require to deal with pair data. Given that the networks are created separately, you don't need to pair your data set before, which is a big limitation in methods based on regression. And then you can get a lot of different types of information using the random walk with restart and multi-x rank from the lab of Anaïs Boudot. We could extract targets for transcription factors, enhancers, so peaks that are associated to the regulation of a certain gene, and so on. The benchmarking that I'm going to show you here is based on the prediction of transcription factor targets, but we also tested the quality of enhancers, etc. So we took the main state-of-the-art methods, Cynic Plus, Serolacol, Pando, and Gene3 just to have an idea of the performances of methods only using single-cell RNA data. And then we compared with the ground truth, which is based on intersection in between perturbation experiments and ChIP-seq, in order to avoid all the possible false positives that are present in each of these data sets. And these are the performances that we could observe. So we are testing it on 4 data sets. The last one is the most interesting one, because it's an unpaired data set. So here, the other methods need to pair the cell and then run the regression model. And as you can see, the black line corresponds to random performances. And as you can see, like all the methods that are pairing the cell fell towards random performances, while we keep quite some stable results. What we are evaluating is the average number of true targets per transcription factor present in the data. And on the x-axis, you have the number of predicted targets that we are taking into account. So we are, let's say, increasing the number of targets and checking the evolution of the right transcription factor targets. We have 2 versions of humus. The yellow one is taking into account also transcription factor cooperation, while the orange is not taking into account this aspect. Given that the alternative methods were not using transcription factor cooperation, we wanted to see whether the advantage of the method was coming from the transcription factor cooperation or more in general from the structure of the network. And what we can see is that both versions perform better than the state of the art, with the yellow one having better performances. So cooperation in between transcription factor as a role for the extraction of the information. The performances, however, are not great. You can see that we have average values of 8, 7, 5, and these transcription factors have an heterogeneous amount of targets, but some of them have even hundreds of targets, and so we are really far from having optimal performances. I forgot to say that that's the PhD work of Remy. And we went then to test the method on a concrete case of mouse cortex, where we had 3 omics data. So we tried to challenge the method to include another types of omics, and in particular single-cell methylation. How we included it? Just building correlation network in between genomics location, and then connecting the genomic location of the methylation with the peaks of the attack based on their proximity, and same thing in between the genomic location of the methylation and the genes in the genome, whether they were close to the transcription starting site of the gene. We could then extract regulons that are set of transcription factors with their targets, and based on the activity of these regulons, we could recapitulate all the cellular heterogeneity that was present in the data, and that was identified using all the 10,000 genes in the expression data. So 10 of 20 transcription factors are recapitulating the full heterogeneity in between the cells. And then we could go to check what were the 5 strongest transcription factors associated to each cell population. And we could see that most of them were already documented to be associated either to the region of the brain where we identified them, either specific to the cortex or to neurons, brain, and so on. So they seem to make sense with respect to that context. And it was interesting to see that all the TF that are indicated with an arrow on the bottom are those that we would have lost if we were not including methylation in the analysis. And so methylation is bringing information, and having a flexible method that can easily include other omics data can make the difference in some analysis. What's the next step? The next step is that here we are looking at the molecular mechanisms that are internal to the cell, but we are not taking into account the fact that a cell lives in a tissue and is interacting with other cells. And so we have on one side methods that are inferring molecular interactions, on the other side methods that are inferring ligand-receptor interactions and their downstream effect, but we don't have something that is putting these two together in order to try to understand, to combine molecular interactions with ligand receptors one. And that's in this direction that we have been developing RECON, which is extending the model that I have been showing you before, still using heterogeneous multilayer network to take into account ligand receptor interactions in between cell types and combining them with gene regulatory networks coming from each cell type in order to model the effect of the alteration of a ligand on a full tissue. And so what we can do with the method is to define the consequences of an alteration of a certain ligand on the full tissue, but also identify possible drivers of a given alteration. And one nice part of the method is that it is combining and weighting two main components, the direct effect that arrives on the cell due to the given alteration, and the indirect effect. So the fact that the cell is not isolated, but is present into a tissue, and so the other cell types, in consequence to an alteration, will send signal to these cells that will further modify its gene regulatory network. And I will show you just one benchmarking result, which is done on the mouse immune dictionary. We have, I think, 17 cytokines that are altered. What we did is to build our gene regulatory network on independent data set, which were as close as possible to this setting, also because we want to have a lot of single-cell data. And then we built our recon network, and we try to make it from the simplest one to the most complex one. So first of all, we can just have receptor to transcription factor correspondences. Then we can introduce gene regulatory network in the analysis, and so have transcription factor gene interactions. Then we can have a ligand that is altered having an effect on the downstream genes, so we can have loops in between the gene regulatory network and the ligand receptor interactions. And then finally, we can have the most complex model, which corresponds to recon, where we are making the gene regulatory network specific to each cell type. And so we are really reproducing the interaction in the tissue. And these are the results that we can observe. This is an AUROC evaluating the intersection in between the prediction that the model is making on the genes that will be altered in consequence of the cytokine alteration and the genes that are differentially expressed in the analysis done experimentally. And what we can see is that these are the performances if you use recon with no context, so no gene regulatory network in the analysis. This is the increase that you get if you add the gene regulatory network. Here, the increase that you get once you allow to have ligand and receptors signaling to the gene regulatory network back, but the gene regulatory network are still not specific to the different cell types. And finally, these are the results that you get if you introduce the cell type specificity. So it's nice to see that we have an increase overall in the performance, is justifying the structure of the network. And it's also interesting to notice that making the gene regulatory network specific to the different cell types didn't make a huge difference as the other steps, indicating that maybe the gene regulatory network that is generic is already not so bad to represent the interactions in between different cell types. So then the last direction is cell trajectory inference. And for this, the idea is that recently we started to be able to access spatial transcriptomics data measured in different time points of the same biological process. For example, here you can see zebrafish development, different phases of the development process, slices of spatial transcriptomics in different time points. And the overall objective that we wanted to achieve was to try to reconstruct the differentiation potential that is behind this temporal evolution. So sort of Weddington landscape that can model the evolution of the cells, having more stem cells on the top with a higher potential and more differentiated cells on the bottom. Dealing with, reconstructing such a potential also allows to make prediction on what will happen to the cells in future time points. While if you're just trying to map cells across time points, you don't have this type of feature, which is drastically interesting and can also allow to model then perturbation effects on a trajectory. What the model is doing is to learn the potential J using a simple neural network, an MLP. And the loss that we are using is minimizing at every time point the difference in between the full gene expression distribution that we can predict from time point t to time t plus tau and the real measurement available at time t plus tau. So you need to have several measurements in order to make a good prediction. If you have only 2 time points, the model will not be able to learn a right potential associated to the evolution. And which loss are we using in order to evaluate the difference in between the real measurement at time t plus tau and the prediction? We are using a fused Gromov-Wasserstein loss. What's the idea behind it is that we want to take space into account and the spatial localization of the cells into account in the idea that cells that are close to each other will influence their fate. And so we have a first term in the loss, which is a linear term, which is taking into account whether the overall gene expression distribution of the cells in between the prediction and the measurement is close to each other. And then we have a quadratic term, which is taking into account whether cells that are close in space in the prediction are also close in space in the actual measurement. And here you have the benchmarking. That's a joint work in between Jules and Gertian, 2 PhD students in the team. The benchmarking is done in 3 atlases. The first one is axolotl brain regeneration. They cut a part of the brain and then they follow its regeneration. Then the second is the zebrafish development, and the third one is mouse development. And we do 2 types of tests. An early test set, where we are taking a different batch from the training data, but on time points that have been already seen during training by the method. And then a late test set, where we are working on a time point that has been hidden by the method during training. So it has never been seen, and we are asking the model to predict its expression profile without having never seen this time point before. Here, what we are evaluating is the velocity time distance in between the prediction and the real measurement in all 3 data sets, early and late test set, and we are comparing stories, which is our method, with respect to prescient, which is an alternative method also based on the idea of inferring a potential for the evolution of the cells, but that is not using the spatial information, so it's just looking at expression data. And what we can see is that overall, the use of the spatial information is making a difference in stories in terms of performances. We wanted then to test whether this was just a matter of distance or whether there was some real biology into that. And in the mouse data set, we have known annotations done by biologists of what are the right cell type transitions in between time points. And so we could compute the transition accuracy, evaluating whether the method was correctly predicting the transitions in between different cell types. And also with respect to these stories as better performances. And then we can go to look at specific applications and test cases. For example, here it's the axolotl development, and we are focusing on adaxial cells. And you can see that stories is correctly predicting them to fit with the bottom of the embryo, while in the case of prescient, the mapping goes to all the structure of the embryo, and this is due to the fact of not using spatial information in the prediction that makes the prediction less accurate. Then we went to check on some real application whether sometimes we could identify a real setting where the space matter. And this is a specific transition in the mouse cortex from radial glia cells to glioblast and neuroblast. So radial glia cells are splitting into these 2 subpopulations. And we were wondering whether the space has a role in this. And what we could observe is that the distribution of neuroblast and glioblast in the mouse cortex is not random, but it has specific patterns. We have on the top neuroblast, then glioblast, then neuroblast again, and finally glioblast. And radial glia cells are distributed all along this structure. You can see the blue cells are in all, let's say, the part that is also violet and green. And the commitment of these cells in this area will not be just related to their gene expression, but will also be related to the place where these cells are. On the right, you have the potential inferred by stories, and we can see that the radial glia cells that are on the upper part will tend to commit to neuroblast. The radial glia cells that are in the middle are red, indicating a commitment over glioblast. Then on the second part, on the bottom, we have blue again, indicating a commitment over neuroblast. And finally, we have red, indicating a commitment over glioblast. So this means that cells, radial glia cells that are in specific locations in the brain will have a commitment that is not just influenced by their expression data, but it's also influenced by their spatial location in the tissue. So with this, I will conclude. I want just to add that we have other work on cell trajectories, not using spatial information, but using metabolic labeling barcoding in order to help the inference of the trajectory and having a local velocity for the cells. So don't miss flow state and the poster of Anodi that is presenting this work. And finally, I want to thank all collaborators, in particular Gabriel Perrier, with whom we did all the part on optimal transport and funding, and the people of my lab that have been doing all this work. It's thanks to them if we have all these results. And thank you all for the attention, and I'm happy to get questions.
Thank you, Laura, for this exciting talk and the development of so useful methods, I think. Are there any questions?
Hi, thanks for the great talk. It was really fascinating stuff. You went on it quite quickly, but in the sprint, I'm not sure how you pronounce it, you mentioned that you were feeding your models an embedding of the conformation of the protein. In what form is that given, and how do you create this embedding of the protein? I'm not familiar with that work.
So we are just giving the results like the output of the SM2 embedding. So we are using like reference foundation models for protein embedding. We are not doing anything special on that side. The idea is just the intuition is that the gene symbol in between the same genes will change across species, and if you are giving to the model data from different species, you will not be able to identify whether it's the same gene. While using the protein embedding, we can allow for that, and this was giving better performances.
More questions? Don't be shy. Yeah.
Thank you very much for the talk. I was interested in the print method. Are the gene recording network you infer directed or just association networks?
They are directed because from the attention matrix you get really whether one gene is looking to the other, so you have a directionality. They are not a transcription factor gene network. They are gene-gene network that can reflect also indirect interactions. We tried in the second work to have also pre-training which is based on masking transcription factors to try to identify transcription factor gene interactions. And that allows to predict better gene regulatory network with certain ground truths like omnipath, but there is not a huge difference in performances in inferring gene-gene networks or transcription factor gene networks.
Thank you.
Welcome.
More questions?
Okay.
Go ahead.
I'm starting. Okay. Yeah. So for the SCPrint foundational model, one of the core premises of all these foundation models is relying on the scalar flow. So the more data sets we ingest, and maybe the better performance. But I know that there's some controversy about this, and some other studies tend to show that it's better maybe to fine-tune to your specific biological context. So what would be your advice if I have a specific biological context, if I want to use SCPrint? Should I use the full database, or should I contextualize, or should I maybe subset only on the top relevant biological data set?
Yeah. So first of all, on the number of cells and impact of that, we didn't see a huge impact on the amount of cells that they are giving in the training, meaning that in cell by gene you have 50 million cells. We don't need to use the 50 million cells to stabilize the training, first of all. And what matters a lot is the heterogeneity of the cells more than the number. More the model can see different cells and more it's learning about their functioning and so on. Regarding the fine-tuning question, I think that every time you use a foundation model, if you have a specific question on a specific tissue, if you fine-tune the model, you are getting more reliable results, especially if you want to get biological insights out of that. So here we were just trying to challenge the model in zero-shot prediction because ideally that's the use that you would like to make of a model in the future. But if right now I have to use the model to analyze data, I would fine-tune it on my dataset specifically.
Hello, I'm here. Thank you for a great talk. I have a question concerning the learning of the sulfates landscape on spatial transcriptomics. You worked on stereosec data. Do you think your pipeline is also working on Murfish or Xenium datasets?
Yeah, so I cannot say anything about Xenium because we didn't test it. Normally, the model is designed for single-cell resolution data, and it takes in input already pre-normalized expression data and spatial locations. So I don't see why it shouldn't work, but I cannot say anything for sure because we didn't test it. For Merfish, instead, we have some tests, because what we are trying to do now is to integrate morphological information of the cells together with gene expression data and spatial location to predict trajectories. And so there we need to work with imaging data to have information on the real positioning of the membrane of the cells. And so I know that on Merfish is working quite well.
Okay, perfect. Thank you.
Welcome.
Hi, Laura. Thanks for the talk. So I have a question regarding the trajectory inference. So because you need this temporal data for the model, so did you look at the dependency of your inference regarding the frequency of acquisition, for example? So how does it depend on that?
So we didn't test this much. It would be indeed interesting to do it. We didn't try to check to which extent the number of time points and association with the biological variability in the system would affect the inference. What I know is the impact of the number of time points, because now we have been using it also with 4 time points for training, and we can still see quite good performances in terms of known transition. So I know that you don't need to have 8 or 10 time points, but even 4 can work. But we didn't do any testing on like extremizing the biological variability in between time points and seeing the effect. We have one test because we tried for a collaboration with the lab in Switzerland. It was zebrafish development, and the time points were reflecting super different time points with respect to the development process, and the model was not predicting anything good. So for sure, you need to have a good, let's say, sampling of the real biological variability and variations that are happening during development, but I don't have any specific information on what's the minimal amount that you need, unfortunately.
Me again, hi. I was curious also about how you're integrating the attack-seek data that you measure, in particular in Hummus, but in the other projects too, if you're using it there. Do you look for do you try and define peaks of accessibility, or do you try more, my instinct would be just to look at 1,000 base pair windows and measure accessibility in each of them and then just see what is in each of these bins, but it's a curious data type to try and integrate. And same with ChipSeek. I mean, the peak information is what's generally used, but it's a very binary type of information that doesn't reflect the whole depth of the experiment in general.
So first of all, let's say the interest of my team, it's really at the level of the integration of the different data types, but we don't dig into the details of the pre-processing of the data themselves. So when we are collaborating with biologists, we interact a lot with the bioinformaticians of the lab that are doing all the pre-processing part and can give us insight on that. But we don't, let's say, modify anything on that part, and we're really working on the part of combining different data types together. For Hummus, what we are doing is to use Cicero, which is an existing model to compute peak accessibility, which is looking at whether they are on the same chromosome, and so close to each other, let's say, in the genome, plus their correlation of activity. For SC-Confluence, we are just using the full peak data set as it comes out of the sequencing. And then what we are doing is to use SIGNAC, which is designed to get gene activity out of the single-cell attack data. So we'll just check whether the genes are close to the transcription site, so whether the peaks are close to the transcription starting site of the gene, and do a sort of average of activity of these peaks in order to represent the expression of the gene. And this is what we are comparing with the single-cell RNA, in the idea that this is not the information that we use for the integration, it's just the information that we use to mix the cells in the latent space. And so it can be super noisy information, it's just an input, a prior that we're giving to the model in order to help it in the integration. Welcome.
And following up on that, on Hummus, so what you say is that the results are kind of limited. So what is missing to be able to perform better? Is it the input data, or is it still method development that is lacking, or what is missing?
What I think is that method development is lacking, but the major issue that we have is that we don't have ground truths that are great in order to evaluate this method. And so this is a bit blocking us from evolving into better methods and having a real evaluation of different methods. I were discussing it before in the context of Sprint. Omnipath and perturbation experiments have a really small intersection in between them. And so, depending on whether you use one or the other to evaluate your method, you can get different methods performing the best. So I think what we would really need is a community effort to design good ground truths and have a proper benchmarking of gene regulatory network methods. And at that point, we would have the elements to try to develop something better.
Any other question? Yeah.
Hi, Laura. Thank you very much for your talk. I have a small question about the integration, your presentation about the single cell and spatial transcriptomic integration.
Yes, and fish and single cell RNA.
Yeah, absolutely. My main question is about the segmentation algorithm that we need to deploy on the images in order to define the cell object. We know this kind of algorithm, the performance is highly dependent on the kind of tissue, if it's the organ, if it's a healthy tissue or tumor tissue, it can be really challenging. And my question is, what is your feeling about the influence of this segmentation on all the workflow and the calculation after.
So that's a real problem. We didn't test much. Like here was just like a fun application for us because everybody is working with proteins, attack, and RNA, and we wanted to do something different. But we didn't test much performances in other tissues. What I can say is that we have been working on stereo-seq data from mouse muscles in Pasteur. And they are like muscle cells have different nuclei, and segmentation algorithm work really bad on those cells, and the data were totally unusable for the following analysis. So for sure, the segmentation part, as you were saying, is crucial, but I don't know what's the impact in the integration depending on the different tissues and so on, because we didn't test it out.
More questions? No? Good. So with this, we'd like to really thank you, Laura. So we will now have a coffee break, and after the break, we'll have 3 parallel sessions. So one here will be about spatial and single-cell genomics. In Amphithéâtre 3 will be knowledge graphs and biomedical data integration. And Amphithéâtre 4 will be machine and representation learning.
If Lucilla Mott is in the room, please come. Ah, je demandais si vous m'appuyez, c'est bon. So we can start. So welcome everybody to the spatial and single-cell genomics session. We start from Adrien Dufour, which is going to talk about spatial transcriptomics analysis, reveals region-specific glial activation during epilestogenesis.
Bonjour et bienvenue. Et du coup, je vais vous présenter un papier que nous avons publié en début d'année, qui est une analyse spatiale transcriptomique dans un modèle d'épileptogénèse. Du coup, déjà un rappel de qu'est-ce que l'épilepsie. C'est une maladie qui est imprédictible, qui est de crise d'épilepsie soudaine, de diverses formes, et qui est causée par des décharges synchrones dans le réseau nerveux. Et chez ces épilepsies, il reste encore 30% qui sont des épilepsies qui résistent au traitement médicamenteux. Et l'épilepsie représente approximativement 50 millions de personnes dans le monde. Du coup, en quoi consiste l'épilepsie? Ça consiste en une première étape d'une première, on va dire, blessure, d'un status epilepticus, où on va avoir un traumatisme et des encéphalies. Puis on va avoir une phase latente qui peut durer de plusieurs mois à plusieurs années, où on ne va pas avoir de crise d'épileptique, et on va avoir un remodelage moléculaire intense du réseau neuronal. Puis enfin, on va avoir ce qu'on appelle une phase chronique, où on va du coup avoir des crises épileptiques récurrentes et spontanées. Et nous, on va s'intéresser plus particulièrement au TLE, du coup aux épilepsies du lobe temporal, qui représente 30% des épilepsies et qui est une des épilepsies les plus communes et qui se caractérise par une sclérose de l'hippocampe avec une perte des neurones dans les régions CA1, CA2, CA3 et DG et une astrogliose. Et du coup, nous, on a un modèle de rat qui mime cette épilepsie du lobe temporal avec une injection de lithium associée à de la pilocarpine. Et du coup, dans ce modèle, nous avons des rats épileptiques avec du lithium et de la pilocarpine et des rats contrôle avec du lithium et une solution saline, dans lesquels on a prélevé les cerveaux à 5, 10, 20 et 40 jours. Du coup, de 5 à 20 jours qui correspond à la période latente chez ces rats, et 40 jours qui représentent la phase chronique. Et dans ces cerveaux prélevés, on a du coup réalisé des coupes coronales des cerveaux auxquelles on a gardé un hémisphère pour réaliser des analyses des analyses spatiales Visium, la version V1, auquel on retrouve à peu près 15 000 UMI par spot. Et le pipeline qu'on a suivi, c'est qu'on a d'abord aligné notre séquençage sur le génome de référence via Space Ranger. On a ensuite réalisé des contrôles qualité. On a ensuite normalisé avec SCT Transform en régressant pour les ADN mitochondriaux. Puis on a intégré en utilisant la méthode CCA qui est inclue dans ce rat. Puis il y a eu une étape d'optimisation du clustering qui a été fait à partir d'un score silhouette et qui a depuis été un peu amélioré et qui a donné un peu le package Narcode que vous pouvez aller voir au poster numéro 81. Et ensuite, nous avons réalisé une analyse des gènes différentiellement exprimés et des voies de signalisation. Puis, nous avons réalisé une déconvolution des différentes populations cellulaires à partir du package CARD. Et du coup, un des premiers résultats, c'est que ici, vous avez notre lame Visium. Et on peut voir que nos différents clusters et sous-clusters reproduisent assez fidèlement les différentes régions du cerveau que vous avez sur la coupe, sur la visualisation de gauche de la laine Brain Atlas. Et on a ensuite été vérifier que du coup ces différents clusters ils correspondaient bien aux régions anatomiques attendues via l'expression des gènes marqueurs de ces régions. Et c'est ici les gènes que vous avez en visualisation, où on retrouve bien les gènes marqueurs du cortex, de l'hippocampe et des différentes couches. Et si on regarde le top 5 des marqueurs de chacun de ces clusters, on retrouve bien des gènes caractéristiques de ces régions. Et ensuite, on a essayé de regarder du coup, qu'est-ce qui se passait dans nos épilepsies en regardant quels étaient les gènes qui étaient différentiellement exprimés. Et du coup, là, globalement, on se retrouve avec à peu près plus de 8000 gènes qui étaient dérégulés, et plus particulièrement dans les régions de la matière blanche et de l'hippocampe également. Et en particulier autour de 20 jours où on a le pic de dérégulation. Et en regardant les voies de signalisation qui étaient associées à ces gènes dérégulés, on a pu observer qu'une majeure partie de ce signal était particulièrement retrouvée autour de 5 jours et qu'à partir de 10 et 20 jours, cette perturbation était plus spécifique de certains clusters de nos régions de nos cerveaux. Et à partir de là, on a essayé de voir un peu plus dans le détail de quelles étaient ces voies de signalisation qui étaient dérégulées dans nos données spatiales. Et pour ce faire, on a utilisé un package qui s'appelle SCPA, pour Single Cell Pathway Analysis, et qui est un package qui a l'avantage d'utiliser une approche par graphe multivarié et qui va du coup prendre chaque gène set indépendamment et qui va, en comparant les graphes entre notre condition épileptique et contrôle, voir les différences et mesurer ça avec une Q-value. Et du coup, cette approche a l'avantage de ne pas se baser sur la moyenne d'expression de notre voie de signalisation et du coup ne pas être biaisé par certains gènes qui auraient un faible niveau d'expression et donc les log functions seraient faibles. Et comme on sait que dans les données single cell et dans les données spatiales, elles sont un peu... qu'elles disposent de beaucoup de bruit et elles sont assez éparses, ça permet de mieux capturer certaines voies de signalisation. Et dans ces résultats, on peut voir par exemple qu'ici, les Q-values sont beaucoup plus faibles finalement à 5 jours et qu'à 20 jours, les voies de signalisation sont vraiment plus particulières de certaines populations. Et ensuite, comme on a des données en spatial, on s'est dit que c'était quand même intéressant de voir spécialement où ces voies de signalisation étaient situées sur nos lames. Et du coup, après plusieurs essais de différentes méthodes, on s'est arrêté sur le package AUCell, qui a l'avantage, c'est qu'il va du coup en single cell dans chaque cellule et du coup en spatial dans chaque spot, ranger les gènes du plus exprimé dans le spot au moins exprimé dans le spot. Et ça nous permet de corriger un peu le biais qui est que certains de nos clusters vont avoir un nombre d'ULMI plus élevé que les autres, normalement notamment dans nos régions C1, CA2, CA3. Et du coup, ces résultats nous permettent d'avoir une approche non liée au cluster, et on aperçoit qu'on a du coup des patchs d'inflammation qui, autour de 5 jours, sont situés autour des régions CA1 et autour de l'hippocampe, et qu'elles vont plutôt former 3 différents patchs par la suite qui vont persister à 10 et 20 jours et qui vont plutôt s'étendre dans la matière blanche et dans le thalamus. Et dans les voies de signalisation qui sont ressorties, c'est qu'on a retrouvé une activation des cellules microgliales et un développement des astrocytes dans ces régions. Et la question suivante qu'on s'est posée, c'était quelles étaient les populations cellulaires qui correspondaient à ces astrocytes et ces microglies. Et là, pour ce faire, ce qu'on a utilisé, c'est le package CARD qui permet une déconvolution et qui va donner la proportion dans chaque spot, parce qu'on peut avoir plusieurs cellules dans un spot en utilisant la technologie Visium V1, et à partir d'un atlas de souris, le Linarson, qui est des souris adolescentes et qui correspondait à peu près à l'âge de nos souris. Et du coup, à partir de là, on a obtenu 130 sous-classes qu'on a ensuite synthétisées et regroupées en familles pour se retrouver avec un nombre de familles plus réduit. Et pour avoir au final à peu près 20 types différents par spot. Et en regardant en global, ce qu'on a pu observer, c'est une augmentation des microglies, et plus particulièrement des microglies activées, et un switch de population au niveau des astrocytes. Et c'est là ce qu'on voit sur les lames Visium ici, c'est qu'en contrôle, on a plutôt des populations de non-télencéphalons astrocytes. Et dans notre condition épileptique, on va switcher sur une population de myoc expressing astrocytes, comme nommé dans l'atlas du Linarson. Et ce qui était intéressant, c'est que très récemment, il y a un papier qui a été un peu caractérisé cette population d'astrocytes myoc, qui correspond donc à une population de cellules d'astrocytes de la bordure et qui était proposé comme faisant entrer le signal d'inflammation dans la périphérie, de la périphérie vers le parenchyme. Et c'est aussi une population qui aurait des liens avec la myélocine et qui serait liée à la différenciation des oligodendrocytes et à la myélination. Et donc, en conclusion, ces analyses nous ont permis de montrer que les packages SCPA et AUCEL donnaient des résultats très intéressants sur les données Visium spatiales. Et d'un point de vue biologique, ça nous a permis de montrer qu'on avait une signature neuro-inflammatoire dans des patchs très localisés, et qu'on avait une astrogliose et une activation des microglies au-delà de l'hippocampe, dans la matière blanche et dans le thalamus. Et du coup, je vous remercie pour votre écoute. Et du coup, si vous voulez voir la suite de nos analyses, c'est que du coup, on a réalisé, comme on avait l'autre partie de notre cerveau, comme vous avez vu qu'on a coupé les lames, on a utilisé cette autre section pour réaliser des analyses single-cell RNA-seq que nous sommes en train d'intégrer et que vous pouvez aller voir au poster 71 de la session B, donc à partir de demain.
Thank you for the great talk, Adrien. It's time for questions. Don't be shy.
Thank you for your talk. I have one question regarding the beginning with the clustering optimization. Have you tried another score than the silhouette one? Because from what I observe with the special data that I have, it's always tend to favorize like a low resolution. So always reducing the number of clusters. So I don't know if you try other score or not.
To be honest, it's not my part of the analysis, so I didn't remember, but I will recommend you to see the poster about NARCode that have been much more developed this part of clustering optimization.
You have a question?
Bonjour. Moi, j'avais une petite question. Encore une fois, c'est peut-être pas les topics sur lesquels tu as plus bossé dans ce projet-là, mais moi, je me demandais en fait à quel point ces conclusions-là se transfèrent à l'humain. Parce qu'il me semble quand même, entre la souris et l'humain, il y a des différences en termes de type de neurones, mais je ne sais pas s'il y a des gènes marqueurs que tu as repérés qui sont conservés chez l'humain aussi, qui peuvent servir à caractériser les types cellulaires et à témoigner aussi de l'impact de l'épilepsie entre les deux organismes.
Alors, pour le moment, non, mais entre-temps, il y a eu une publication où ils ont fait du single-cell RNA-seq sur des cerveaux de patients. Et du coup, l'idée, c'est, avec nos données single-cell RNA-seq de NORA, de faire une comparaison avec les données humaines. Et c'est dans la continuité du projet.
Other questions? One curiosity on my side, maybe I can, and do not hesitate to reply in French if it's easier, I'm going to understand. Did you try to look into ligand-receptor interactions, variability in space, especially during the process, whether there is some variability in the ligand-receptor interactions? And the second question, did you try to look into spatial domains? So to try to combine the spatial positioning, let's say, of the cells together with the expression, in order to see whether you can get some more fine variability in between the different conditions.
For the ligand receptor approach, we were waiting for the single-cell RNA-seq to have a more deep in the transcriptomic part. And for the second part of your question, no, we do not have a look at this for the...
Okay, thank you. Other questions? Okay, if not, there is a question there, up on the left. The microphone is arriving.
Hello. So I have a question. You worked on the rat model. Is there any reason why you didn't use the mouse, as Allen Brain Institute made the reference with the mouse brain?
I didn't remember why, but I know it was the better was better with rats than mouse. And,
okay, so did you encounter any problems when using your pipelines on rat models?
For the alignment and the sequencing, for the alignment of the rhythm, it was done on the rat reference annotation. And for the deconvolution with the Linarson, we was a little bit. It was difficult because we have difference if we use the Linarson on the Allen Brain Atlas, but it's both a reference of mouse. So there are already difference between mouse, so that's why we want to reintegrate with an in-house single-cell RNA-seq with rat, because, for example, the myoc astrocyte subpopulation was not present in the Allen-Bowen atlas, so we can't find it in our data.
Okay, thank you.
Great. I think it's time to move to the next speaker. Let's thank again Adrien. So our next speaker, while they mount the presentation, is Gianni Zanardelli, who is going to talk us about spatio-temporal regulation of cell cycle states within the complex tumor microenvironment. And the floor is yours.
Thank you. So, oops, sorry, I can start, yeah. So hello everyone, so my name is Gianni and I'm a second year PhD student and I will talk about part of my thesis project done in the lab of Nacho Molina in the IGBMC and now in Madrid as well. So, okay, this is working like that. So yeah, quick reminder about cancer. So fuck, it's weird to do that. So it starts with cells that are genetic mutation and they start proliferating without control, and in the end we have tumors that have the possibility to invade other parts of the body. This proliferation is due partially to the misregulation of the cell cycle. So basically we have classically described it with different phases such as G1 where the cell grows inside and fulfilling function, the S phase where the cell replicate the DNA, and the G2 and N phase where the cell prepare and divide into two daughter cells. But even if we describe it this way, it's a continuous process, and I call it theta. So when I will talk about discrete phase, I will talk with G1, S, G2, and M, and when I talk later about continuous cell cycle, I will call that theta. And cell can also exit temporarily the cell cycle and become what we call quiescent or zero, which is also a big issue in cancer because, okay, it can stop the proliferation if you don't divide, but then it is not focused generally on the therapy, so it can provoke relapse years after the cure. And I won't talk much about that, but cells can also totally exit the cell cycle and become senescent, and this plays also a role in cell cycle proliferation in cancer. And so the other main, I'm lost there, part of my topic is the tumor microenvironment, which is the complex ecosystem of cells inside the tumor. It's not just made of cancer cells, and it's made so with immune cells, endothelial cells, stromal cells, and cancer cells, and all these interactions can also influence the proliferation of the cancer cells, so there is a link between cell cycle and tumor microenvironment, and that's what I'm going to study there, thanks to spatial transcriptomics. So for those who don't know spatial transcriptomics, here I'm using Mayfish data provided by this gen company, so it's publicly available data. So on the top left, you can see in blue, so it's a tumor section, it's a special one. You have the TAPI for the cell nuclei. In yellow, you have some cell boundaries. And all the small dots you can see there are currently RNA that are probes there, and so observing the data. And if we zoom out, we have, for example, it's a lung sample tumor, so you have the scale, so it's almost 1 centimeter square, and here we have almost 400,000 cells and 500 genes labeled in each cell, so I have around 100 million of transcripts that are labeled there. And once I have the data, because it was not provided, sadly, I have to identify the cell type and doing the clustering, so I'm doing classical Leiden clustering and then looking at the top genes for each cluster to identify the cell type. And so I have different tumors there. So you have the special plate at top with melanoma, breast, and lung that are provided by the company, and the UMAP on the bottom. In blue, you always have the cancer cells, and what you can see is that we have different cancer cell population, especially in lung where it's quite visible. In purple, orange, and so on, we have the immune cells. So we have, for example, infiltration of immune cells in some part of the tumor in the melanoma, and it's a little bit scattered. Sorry, it's still complicated to do that. On the other tumor. And so it was just to check the spatial distribution of the cell type there. And for the rest of the talk, I will focus on the lung, but then the idea is the same, and you will be able to see the same story for the other data at the end. So yeah, just to better visualize the cancer cell distribution in the lung, so you can see that there is kind of an onion shape with 3 populations, and one is surrounding the other one, and the last one that is the light blue that is surrounding everything and the most present. So I have 2 questions there for now. So now that I have the cell type, I can find the cells that are in the cell cycle and then the cells that are not in the cell cycle to do further analysis, and also check what can explain the distribution of the cell the cancer cells, how they were divided into different populations. So first, using existing method in ScanPy, so I'm doing Python there, with the cell cycle score genes, I have some gene markers for each phase, G1S, G2M, and G0, and then doing that, I can identify the phase of each cell type, sorry. And what you can see is that, for example, in yellow, it's the cells that are quiescent, and they are also strongly located, not randomly distributed in the tumor, and same for the other cell cycle phase, and just a comparison with that. And if we look at the cell cycle phase distribution among the different cell types. So we have mainly majority of the fibroblasts and endothelial cells that are quiescent and not proliferative, which is expecting and none to not cycle. And what is interesting, it's for the cancer cells, we have the cancer cell 1, so the light blue that have more than 100,000 cells that is highly proliferative, with half of the cells being in S phase and 30% being on G2M. While in G2, in cancer cell 2, it's half of the cells that are either in S or G2M, and cancer cell 3, it's even like less than 40%. So there's the clustering already identify cell cycle differences between the different populations of cancer cells. And then I also do some gene set enrichment analysis to identify differences between the different populations of cells. So the cancer cell 1, it's only cell cycle pathways that are enriched. So I'm using the Reactome database. And then for cancer cell 2, it's a little mix of everything. And cancer cell 3, which is the less proliferative one, there is a lot of response to stimuli or stress and some events that can explain why this population is not proliferative and mainly resting or in G1. And yep. Then, I have another question was to see what can explain also, especially speaking, the differences, why there are some cells that are quiescent and some others that are proliferative. And the first idea I have is that, okay, these cells, the quiescent cell may be in high dense regions, like surrounding by a lot of cells and cannot have access to nutrients, for example. So for this, I just take every cell, I put a radius of 50 microns surrounding it, and I count the number of cells surrounding every cell to have the density, the local neighborhood density for every cell, and this is what I show there. So there is no differences between all the cell cycle phase. For the other tumor, there is quiescent cells are located in less dense regions. And then I group the cell cycle cells together, and I looked now not by just the number of cells that are surrounding every cell, but if they are quiescent or proliferative. And what we found, and that's what I have for every data, is that quiescent cells are located in quiescent region where a lot of quiescent cells are surrounding quiescent cells and less proliferative one. But if I'm a proliferative cells, I will be surrounding by other proliferative cells and not quiescent. And then I looked also with all the cancer cell population, the composition of their neighborhoods. So I grouped by cancer kind of compartment, so cancer cells, immune cells, stromal cells, and endothelial cells. And what changed the most between all the population is the enrichment or the differences in the immune population that changed most. So, and the stromal cells, so there is way more stromal cells surrounding the cancer cell 1, which is the most proliferative, while there is more immune cells surrounding the less proliferative cancer cell population. And now that I have done all these analyses on the, let's call it the discrete phase, cell cycle phase, I want to go only with the proliferative cells and go deeper into details and look at the continuous cell cycle. So to model this cell cycle, I developed an expectation maximization algorithm. So it's a two-step iterative method. So on one step, I have my data. So on the y-axis, you have the gene expression level, and so you have the cell cycle on the x-axis. Each dot represents a cell, and so you put the cell according to the expression of the gene, and then you model the dynamic of every gene expression. And if you have the gene expression dynamic model here, you have to put the cells at the correct position of the cell cycle. So you will scan all the possible cell cycle value, and you will end up with a probability distribution for every cell along the cell cycle. And the highest probability is more likely the position of the current actual position of the cell in the cell cycle. But we don't end up quickly with the perfect solution, so we'll go back and forth between these two, with having a new probability distribution for all the cells, and then we will map the model according to that, and then we calculate new probability according to the new model, and then we will reach convergence. And this is some of the results I have with two cancer cell population there. So with the cancer cell 1, the most proliferative, on the top you have the proportion of the cell cycle, so S in red, G2M in green, and G1 in blue, sorry, along the cell cycle. So here you can see that these cells are 50% of the time in S phase, which is also the phase with the less variability on time, and then they spend 40% of the time in G2M, which means that this population is really highly proliferative, and there is almost no G1, so they will go into S, then divide, and then directly replicate the DNA after, and doing that for some time. And on the bottom, you have the distribution of cells along the cell cycle. So there is some technical issue with the EM algorithm when it finds some peaks where cells are mainly grouped, and it's so technical issue, and due also that I have only 500 genes and among 20 genes that are cell cycle markers, so it's not a lot, and it can be complicated to do simple things with very few data, and also the quality of the data also is not as good as single cell RNA sequencing. And so for this population of cancer 2, which is the intermediate proliferative, so cells spend more than 50% of the time in G1, which is also the phase that have the highest variability on time. So these cells are way less proliferative, and they spend like 25% on S and 25% of G2M. So these cells are, this population is way less aggressive than this one. And this is from the cell point of view, but then we can also have the gene point of view with the gene dynamics. So here we have just a neat map showing the variation on time, so during the cell cycle of some genes. So when it's red, it's upregulated. When it's blue, it's downregulated. And first to see also if I was doing crazy thing or not, I just check the cell cycle markers I've used, like MSH6, MCM2, or PCNA for the S phase, and they are mainly expressed so during part of S phase, which is nice, and same for some mitotic markers like BUB1, AURKA, AURKB, FOXM1K67 that are expressed during G2M phase. And what is nice also with that is that depending on the cell cancer cell population, we can find genes that are not associated with the cell marker, but that will express an oscillatory behavior. So here we have, for example, SOX2, which is also associated with plasticity. So this cancer cell population is quite aggressive, and it's also can maybe not becoming cancer stem cells, but they start to express some plasticity, so it will be more complicated to fight against this type of cancer cells. And also, like, check to LGR5, I haven't looked at all these genes. There is so many genes to know, it's complicated. And same for the cancer cells 2 population. So you can also see a limit with the data I have, is that there, S markers are also highly expressed in the G2M phase, so there's kind of overlap between the two phases, so it's more complicated to differentiate the two there. But there is also some other non-cancer cell genes like SERPIN1 or that is expressed FN1 as well. And some developmental genes like VEGFA, which is a non-angiogenesis growth factor. So that can lead to the formation of endothelial cells. So this cell can be less proliferative, but also acting in the tumor growth by doing other things as well. So this is interesting to see how non-cycling markers are behaving in the different cell population. And so yeah, you can see all these stories. So the preprint is now online. Just check this morning, the code is private, so I have to put it public. I will do it this week, just doing that. And I'm also doing kind of revision on it. So yeah, doing revision. I'm applying on more data as well. And yeah, thank you for your attention. If you have any questions.
Yep. Thanks for your talk. It's time for questions.
So you said that you used the EM algorithm to retrieve the cluster the the temporal dynamics of your cell cycles. Which model have you used in the EM algorithm? Because the EM algorithm is just a technique for complex optimization problems, but have you used simple Gaussian mixture models, or have you tried to use a specific model that would enable to account for the temporality of your dynamics?
Well, it's something that we develop in the lab, not based on something that already exists. So in the techniques, we have the cell cycle as a latent variable. And so based on that, and we use also an uniform prior for not infer anything special and just see how it evolves. And for the model of the gene dynamics, I'm using Fourier series. So I'm going between the Fourier modeling and this likelihood with the cell cycle as the prior.
Okay.
Hey, thank you for your talk. I think it was very interesting. Oops. It's fine, like that.
Maybe there's
very interesting work. I was wondering about the slide you showed on the cell types and the proportions of each cell cycle.
Yeah, this one?
Yes, this one. So this is something that you inferred from your data, right?
Yeah.
I was wondering if it's like typical image of how this should look like, or there is something like, so should I expect it in every cancer, or do you have any idea about that?
For some population, like fibroblasts and endothelial cells, I expect them to be not in the cell cycle, or the majority of them not being cycling. But then for the cancer cell population, I was not directly expecting to have one population that is highly proliferative and the other not. But also the thing is that I don't have a lot of genes. That's something we have to keep in mind with only 500 genes. So there is some also variability that have to be taken into account. And then for the immune cells, I know that there are populations that are proliferative, some others. So, but then in proportion, I don't really know for the immune cells. And this is something I observe with all the data I have. So.
We have a question there.
Hi, thanks for this. It's a sort of related question. So here you say you distinguish these three cancer cell types. Do you think is this like characteristic of this type of lung cancer? Or would you, or in the other tumors, would you expect to find 2 cell types or 10 cell types, or?
Well, the thing is that I know that is a lung tumor. I asked the company which type of lung tumor it is, but they never answer me. But then it's also depend of the resolution you choose for the clustering. You may have more or less. So we try to keep something with not a lot of cluster, and also doing some scoring to characterize. And with all the data I have, I always end up with 2 or 3 cancer cell population. So I have sometimes cancer stem cell population, but with what I'm currently doing and how I'm doing it, I'm always end up with between, let's say, 2 and 4 cancer cell population.
And I was also wondering, so you did this all on this one data set, and
Yeah.
would you expect to find similar results in other replicates or in different tissues or things like that? I don't know how much you've investigated this or if you're planning on digging these further.
So I don't have a lot of data. So in the preprint, I also have the breast and the melanoma tumor that I presented in the beginning, and I have similar results, but with special differences. And I'm currently also doing it on other data that I found online. So to do, yeah, let's say high statistics, or I don't know how to call it, it will be take some time, but I expect to have some similarities and some differences between all the tumor.
We have a last question there, the top left.
Hi, thank you very much for your presentation. I was wondering, did you pick the 3 cancer cells according to different spatial regions of this tissue, or was the question of the spatial, how do you call influence of their apoptosis?
No, for the clustering, I just took a normal Leiden one. I know there is now clustering that take into account the spatial information, but I did not use it there.
Okay.
So it's purely on the transcriptomal similarities.
Okay, thank you.
Great, let's thank again the speaker for the presentation. And we move to the last talk of the session. That's Lucie Lamotte, a consensus-driven framework for building and sharing single-cell atlases applied to pancreatic ductal adenocarcinoma. Let's see if everything works. Great.
Hi everyone, I'm Lucie Lamotte, and today I'm going to talk to you about, is the mic like this okay? Okay. Single cell atlases in pancreatic cancer. On the agenda today, first I want to focus on our case study and then take a look at what already is available in the state of the art and how they build their atlases. Then we'll move on to our atlas and how we do things a little bit different. And then we'll check some of the application for such an atlas. Let's get started. Our case study is pancreatic ductal adenocarcinoma, also known as PDAC. It is one of the most aggressive malignancies with very poor therapeutic option and a very low survival rate. It is a very complex tumor that is characterized by a very dense and heterogeneous microenvironment. As you can see on the figure here, there is quite a lot of difference between the cancerous tissue and the healthy tissue in its composition and organization. Here they focus a bit more on immune cell types. In our groups, we focus a bit more on cancerous cell type, such as basal and classical cell types, the classical one being a bit more differentiated and a bit less aggressive than the basal one. As I mentioned, it is very complex, so it makes it very challenging to study. And we're not the first one that thought about creating an atlas with single cell. When researching the literature, I found quite a lot of atlas mentioning creating an atlas, but then the article that really made it available, we only have 2 atlases available. They have quite the same strategy for building the atlas. First, they integrate for batch effect correction, and then they annotate the clusters manually with few markers. There are some limits to that approach. When integrating early on, you risk losing some interesting biological data, and you risk also the dominance of your larger data sets. When annotating manually, you lose reproducibility, and you also have a limited robustness of your markers. So today, of course, I will try to sell my approach to you. I don't know if it's better, but it's different, so You'll be the judge of that. Our approach tries to be a little less biased in its cell type annotation process. We also try and prone a quality over quantity approach, and I'll come back to that later on. We also went for a late integration strategy. All right, with that in mind, let's take a look at our atlas. First, I want to introduce our input data. We have two types of input data. We have our single-cell RNA-seq datasets and some marker database. For the single-cell RNA-seq datasets, I retrieved 18 datasets from publicly available database. They have both cancerous and healthy tissue samples, and all of them coming from various studies. We spent a lot of time normalizing the metadata. If you know the struggle, you know. Then all of these datasets went through a pretty standard preprocessing, removing putative errors in sequencing and dying cells, for example. And then after putting aside some test cells for testing later on, we end up with approximately 800,000 cells with all of the datasets. That's for the single-cell RNA-seq. For the literature-curated database, we selected 35-ish markers, cell types that are specific to PDAC, and then we search in the literature for signature for such markers, for such cell types. And we organize the cell type in this hierarchical tree of cell types, and we end up with approximately 1,400 markers. If we zoom a little bit on this figure, that was not quite readable, you can see that does that work? In level 1 for epithelium, we have a broad cell type, and as you go further down your cell type tree, you will get more precise cell types. This is it for our input data. We have single cell and marker database. And now we move on to, for me, the tricky part of this atlas build, that was the cell labeling step. We decided to went for a reference-based cell labeling using the literature-curated marker database I just presented. We also went for per dataset annotation instead of integrating early, using several methods. And then we reassembled all of this information into a consensus annotation. Just a side note on some of the bioinformatic technical choices that we made. We used a SnakeMake to build our workflow for reproducibility purposes and also to manage all of these data sets and scripts that was quite hard to do otherwise. And we went for only R-based tools. For the cell labeling tools, we settled for 5 of them in the variety that are available in the literature. We decided to go for quite different tools in their algorithm itself, but also in the way they are sort of marketed, sometimes as signature scoring tools or as single-cell labeling tools. They also differ in the assignation granularity. Some of them annotate at the single-cell level, other at the cluster level. So for all of our datasets, they were annotated using these 5 tools, hierarchically following the hierarchy I presented just earlier in a cell type. Here's just an example for one dataset for the first level, so broad cell types, of the annotation. This is a UMAP representation for all of the 5 annotation tools. As you can see, they don't totally disagree. You can see some similarities, but they are different, and it's hard to choose which one's right. So we decided to leverage that. I'll come back to it in a sec. Just to sum it up, in case I've lost some of you, I'm the last one before lunch, don't worry. So we have 18 datasets that are all hierarchically annotated using 5 different annotation methods. That is a lot of information, a lot of data that we reassembled into a consensus annotation. This is where we went for our quality over quantity approach. By doing a consensus annotation, you of course lose a lot of cells, but we want to think that our cell types are quite sure after a consensus annotation. Let's say, for example, that this is our a simplified version of the cell type tree I shown you earlier. Each cell is annotated with the 5 annotation methods and can fall into 4 categories. First one, no consensus is reached because the methods just don't agree. Then you can have 3 types of consensus. The first one is a full consensus where at least 3 methods out of the 5 agree on a precise cell type. Here, for example, cell type D will be chosen as our consensus annotation. Then for the partial and ancestry consensus, this is where we utilize the hierarchical structure of our annotation. For example, let's say here, the cell type D was found by one method, and then the other methods did not quite manage to get as precise, to go as far in their annotation process, but they're still in the ancestry path, I called, of the cell type D. We'll say that even though they don't totally agree, they don't disagree as well. So good enough, they support the annotation of cell type D as a consensus annotation. For the ancestry consensus, this is where 2 methods don't agree on a precise cell type. We don't know which one to choose. We'll come back a level here, annotate cell type B. For all of these 3 types of consensus, you need to have at least 3 of the 5 methods that maybe don't agree but don't disagree. Okay, now let's take a look at this process applied on our data sets. This is a representation of the dataset contribution to all of our cell types of interest. Here, I only kept the leaf cell type, the precise one that interests us. I want to point out 2 things. First, that using datasets separately, you cannot have all of the cell types that interest you, so it is really by compiling all of the dataset that you manage to have such variety of cell types that can be interesting for an atlas. And as well, as I mentioned, we are interested in classic and basal cell type, and we managed to have quite a lot of information on these cell types, so we were quite happy about that. Now, even though I'm fairly convinced by our method, it's hard to tell if we did things right, so we tried to sort of evaluate what we did. So we did a late integration. I mean by late, like after annotation, we integrated all of our data using Harmony. As you can see here, the dataset are quite well integrated. And when you color it by consensus annotation made prior to annotation, you can see that the cell types, the cells that were annotated with the same cell type do cluster well together, so that was quite reassuring. Then we also took a look at some markers that were used in the atlases I mentioned earlier in this presentation. This is one example of on one dataset of 2 markers, but it was pretty convincing that the right markers do light up in the right cell type, so ACNR lights up ACNR. That's reassuring as well. With all of that work done, we aim to provide to the community all of our work, so that is soon to be completed, I hope. We want to provide the literature-created marker database, and we also want to provide some newly identified marker. This is still some work in progress. We are trying, as well as a pretty standard SORAT process. We are also collaborating with statisticians in Lyon that are working on a new tool that is canal-based to identify markers and outliers. We also want to provide our individually annotated data sets with all of the detailed metadata and integrated data set. That is also a work that is still in progress for the integration methods. And our code is already and will be available on GitHub. I'm reaching the end of my presentation. I just want to focus on application, like what can we do with such an atlas? What's the point? There is 2 sort of application for that. You can, the one that comes to mind first, at least, you can use the atlas to annotate new single cell data using transfer learning tools, for example. SARCHES is a pretty standard tool. You can also use it to deconvolve mixes for bulk RNA-seq or special spots of Visium technology. And this is what we do the most in our teams. So, for example, this is a work of Mehdi Marchand, who is a PhD student. He will be using the atlas to deconvolve bulk RNA-seq data to study the impact of treatment on different cell population. Mehdi and Luis will also be working on some deconvolution of spatial transcriptomic Visium spots to characterize the spatial heterogeneity in pancreatic cancer. Just a quick advertisement. Mehdi is presenting his poster tomorrow, and he's studying the intermediary cell state in cancer cells, so feel free to go check it out tomorrow. Now I want to end on some open question, just to open up the discussion. If you want to talk later on, I will be very interested in your opinion on this question. For example, what do you think is the best format for sharing single cell? There are quite a lot available out there, and they all have their cons and pros. What do you think are the best integration methods for deconvolution purposes that don't only have embeddings? And also, what do you think could be great complementary strategies to validate an atlas, for example? This is the end for me. I want to thank you, JOBIM, for allowing me to speak here today. I want to thank my team and my main collaborator, Yuna and Magali, for their partnership, the rest of my lab, and all of you for your attention. Thank you.
Thanks for the great talk. It's time for questions. We have two there.
At which point, at what extent you've integrated your dataset because and you homogenize your metadata? Because one of the issues when we integrate all these datasets is that potentially we observe different pre-processing. So have you started with the raw counts, or did you try to apply the same pre-processing pipeline? What would be your recommendation for this?
We try to start with the raw counts as much as possible. Sometimes the papers say, like, this is available there, and you are given a count, and you hope that it is unprocessed. But the truth is that I'm not 100% sure, but I did my best, and so all of the dataset went through all the same pre-processing, hoping that there were raw counts, but that is a blank spot on our side.
And also, is it possible with your pipeline to detect outliers or cell types that do not belong to previously annotated cell types?
For now, no. We are really focusing on our markers to annotate our cell type because the tools allow for cell type signature and various markers. It's not just all random, so it's only the one that we selected that can be annotated. We still have a lot of cell types that were not annotated, so we could go back on that and keep all of the cells that weren't able to be annotated and then do maybe some more exploratory work, but it was not the primary focus for now. But we have all of this metadata and data, so will be used, I hope.
Thank you.
Thanks.
Hi, thanks for the talk. It was really interesting. I was wondering if you could detail a bit more your the way you select a cell type in your path consensus, I think you described it as. So when you have different models that will predict different cell types that are still in the same continuity, is there a weighted decision method to try and select one of these different stages that is a possible annotation for this cell?
I'm not quite sure I understand your question. Can you elaborate a bit? Is this what you want to base your talk on?
So for example, in your partial consensus where you have one model that predicts cell type A, one model that predicts cell type B, and one model that predicts cell type D. Here you've said that you're selecting cell type D, but do you have any way of knowing that model 3 is more confident in cell type D, or that maybe the truth is actually cell type B and that it's just that model 3 went a bit too far in its predictions, that sort of idea?
I don't have any way of knowing that it's true, so we assume that it was more a lack on the capacity of the tools to get as precise, because for cell type, for method 3 to annotate cell type D, it first annotated cell type A, then B, then D. And the fact that you could say that it could be cell type B, but in our case, for example, let's say for the basal and classical cancer cell type, the third level would be classic type A or classic type B, and the level 2 would be classic. So it's not contradictory, like we're still annotating it as classic, is same for macrophages, for example. You're annotating it as a precise type of macrophage, but not the broad one. So I feel like keeping cell type D is not really contradictory to cell type B, but we have all of the detailed metadata, as I mentioned, and for now, I decided to keep, for the purposes of the PhD student, for example, the precise cell type of classic type A, type B, such, and so on. But you could decide to come up a level and keep only the gross cell type and be less precise, but potentially less prone to error. It could be a choice, and I will try to really detail my metadata. I'll do the work, promise.
While I have the microphone, maybe if I can, I was also, maybe I just missed it, but this hierarchical annotation, did you do it just for pancreas cell types, or was it a more
This is
global
the issue for now. Our workflow is pretty tailored to pancreatic cancer. I decided, so this is sort of hard-coded, like the whole process is based on the literature-created marker database that I created. So the idea can be used on other types of cancer or tissue, but for now, the process is really tailored to the cell type we chose. So,
thank you.
No.
Thank you for your talk. I also have a question regarding this. Are you, or is it possible to also not only have the ancestry tree, but also having the transdifferentiation process that can happen, or
the what process?
Transdifferentiation. You know that you have one final cell type, but that can go into another one without having the normal path of differentiation, or.
Do you mean if we have a more detailed cell type of transitioning in between cell type? Is that what you mean?
Instead of having like a tree, and you have also like branch that link to final state, or.
Yeah, no. For now, we don't have that. It's like I said, all of the tools are based on a cell type having a signature, and so we don't have this sort of in between, in middle. This is a limitation to the approach, of course. We don't went for a general pathway, just cell type signature, pretty not continuous, the opposite. Don't remember. Discrete. Thank you.
So thanks again for the talk and for all the speakers of the session. That's the last talk. Just a reminder, it's not yet time for lunch. We have a poster session until midday and 2 demos. So take advantage of this time to discuss in front of the posters with the different people presenting a poster, and also for the speakers. I know that there were some open questions that could not be asked now, so take advantage of the break to discuss.
Hello. So, almost everyone is seated, so we are going to start this session. So it's my pleasure to welcome today Christophe Decimos. So Christophe is professor at University Lausanne in Switzerland. He's group leader at the Swiss Institute of Bioinformatics, so SIB, and he's even the director of SIB. So maybe for the young ones, actually SIB has been really a beating heart for a long time of bioinformatics in Europe. Even when I was a student, using the online portals like Expazy or the database Uniprot, I mean, come on, who has never used Uniprot here? It has really always been a big resource of tools and databases in Europe. So it's no surprise that SIB has been now the Elixir node of Switzerland. And Christophe is therefore also the head of node of Switzerland, and is actually very active at the European level. And just for the comparison, in France, it's the Institut Français de Bioinformatique that is the Elixir node. So today, Christophe is going to present how open databases in biology can be strategic infrastructure. So the stage is yours.
Thank you so much, Marco. Thank you to the organizers. Thank you all for being here. It's the second time I'm at JOBIM. I was in 2019 in Nantes, and it was really fantastic. So when I had the invitation, I really could not pass on the opportunity, and I think it's a very special conference. Also, want to particularly kind of engage also with all the early career researchers in the audience, because I think this is a conference that has a particular level of energy. And I hope that, well, I'm pleased that you came back after lunch for this talk. It is sometimes maybe when you're still like doing your master's or PhD, you may wonder, okay, you know, do we care about infrastructure, competitiveness, or sovereignty? But I hope that you will see that in the talk that this is something that can be directly relatable. And so, in fact, I want to start by getting a bit of a sense in the room what role do you see for yourself as bioinformaticians? And so I have 4 categories here, and I will ask in a second for you to pick one. The first one is your main motivation will be to uncover new biology, so discovery. The second one would be to develop cutting-edge methods, maybe some new algorithms, some new methods, or perhaps also benchmark the methods. The third one would be to enable others to do perhaps the first or the second category. So maybe you're doing a lot of training, you develop some databases, some infrastructure, some software that is reused a lot, and you're basically enabling others. And then I added as a 4th category for those of you who feel that it's just impossible to identify with just one of these categories. You think what you're doing is actually sometimes one, sometimes the other, at least 2 of these categories. So I hope this is clear. So I'm interested to know who identifies with the first category. Please put your hand up. Okay. Okay, the second. Okay. The third category. That's very interesting, actually. More people than I expected. So, of course, for you, this talk is directly relatable. And then how about the 4th category? Okay, that is also very, very impressive. There's sometimes a tendency to pigeonhole ourselves into one of these different categories. Nevertheless, I think it is a useful framework, and I hope you will see in the discussion why I'm asking this upfront. And so I have another thing that I would like to ask, and in fact, we already had a bit of a preview to that. I'm curious to know, in the last month, how many of you have had to use an open database, data resources such as Uniprot, String, but also, you know, we have some excellent one in France. So in the last month, yeah, okay, that's many of you. Okay, that tells you already why the discussion we are having now is relevant. Now, of those who had their hand up, how many know that the database that you're dependent on will still be funded 5 years from now? Okay, we have some optimists at the back, but not that many. But in fact, it is a real question, and it's quite paradoxical because, I mean, these databases have been, you know, are kind of more used than ever. I see lots of people at the back. Actually, there's lots of seats in the front if you want to come in the front, and it's also a bit cooler in the front if you like to come down. So, yeah. So, I will go a little bit more into the role of databases, but also just kind of a bit as a warm-up. It's also interesting, and we will discuss quite a bit the role of these databases for research and for these different activities that we've discussed. But actually, I would argue that even with the pandemic and with some other kind of societal challenges, there's also a realization that these databases have a relevance beyond research. And so this is a picture from around 2020, you know, in front of the parliament in Switzerland, and you know, it says that everyone has to stay at home. I'm sure that many of you still feel a bit scarred from this experience. I do not want to bring too many bad memories, but it's interesting to remember what happened in terms of then the kind of the scientific response to the pandemic. I mean, first, the sequence of the virus was shared by some Chinese teams in early 2020. That was shared on virology.org. And so immediately, if you have a sequence on its own, it's not very useful. You need to be able to relate this in the context of what is known. And indeed, I mean, it was very helpful to have already quite a bit of biological knowledge about the 29 proteins of the SARS-CoV-2 virus. Within weeks, people started modeling the spike protein. So the AlphaFold was not available to the extent it is today, but nevertheless, there were already some resources that were used to model the structure of the virus. And then, of course, this data had to be collected, so there were parallel efforts done in different countries. In Switzerland, we had the Swiss Pathogen Surveillance Platform that had been developed to collect bacterial data that could be repurposed on a short term to share this data. And then we had some tools that had been developed to genotype mixed population of viruses, in particular HIV, that could be repurposed also to analyze the sequences that could be gleaned from wastewater. And you remember also how we could track the progression of the disease using resources that basically tie the geographic location and the phylogeny of the viruses, and was helpful also to identify the different variants. And so these I've only highlighted here kind of SIB resources that had, by the way, all been kind of supported and developed long before the pandemic, but that were critical then for this rapid response. And so I think this is a kind of an interesting, also, like very concrete example of how this work that is being done in the background for many, many years all of a sudden can have an extremely kind of relevant impact for society. And of course, today we talk a lot about AI and about the importance of data. In fact, it's not just important for the JOBIM community. It's also important to Ursula von der Leyen, apparently, because a few years back she had a speech in Davos actually explaining that the data is like the new oil, but it's a renewable resource, contrary to oil. And researchers can mine this data and find some hidden gems, hidden treasures, and extract some new insights, not just from their own data, but from data from others. Which just shows already a couple of years back, there was already a sense, okay, there's really something important. And obviously, it's only gotten kind of more relevant. So what is the, I mean, if I take a step back, what is really the value of kind of open science? I mean, you all know that. I know I'm preaching to the choir, but it's helpful to have this framework, I think. We have individual researchers or clinicians generating data in the context of relatively specific projects or efforts, and then by depositing this data into databases and organizing this data, we can hope that this data will be reused and reanalyzed by others, but not just the people inside, that are directly tied to the generating labs, but also the research community at large, and also I mentioned, the government, industry, of course, is using also a lot of this data, clinic, public health, and the public, and of course, AI. And so this is what has also really given a lot of leg to the, the efforts from funders and institutions to push for fair data, because if you have fair data, then this, by being findable, accessible, interoperable, and reusable, the data can be then effectively reused. But actually to do fair data, it's a bit like if you're the coach of the French national team and you say, tonight our goal is to score more goals. It's a good strategy, but it's usually, I mean, unless you're the French national team, it's not always easy to then implement. And so how do we actually achieve fair is a real question. And so I think rather than seeing this as a goal, I think it's sometimes helpful to think of the process. And actually the process that we have, again, is from a very specialized context into a very general and interdisciplinary context where you can no longer assume that the expertise and the specific information that you had in where the data originated is available. And AI is, of course, a very good example of that, because it is, in some sense, like the ultimate non-specialist. It needs to be fed explicitly with all the prior data and knowledge. So I think it is helpful to think of this as a data democratization process, and to think, okay, democratization also, it's a bit of an open-ended goal, but in that sense, it maybe helps us a little bit better to prioritize our efforts. We should think, what sort of effort we can have that will enable taking these kind of very specific data and make them really broadly useful. And when I talk about data in this talk and more broadly, it's in a very general sense. It encompasses both, from the raw data. Usually when people conceptualize the pyramid of knowledge, you have the notion that you first have the raw data, then this is kind of being processed into information and then distilled into knowledge. Sometimes we hear even about understanding as one more level. But in any case, I think it's good to remember that when you need to transmit data outside of your subfield or to the machines, you also need to encode explicitly knowledge. So all of that has to be encoded. So there's maybe a little bit of knowledge that I will transmit today in form of speech, but it's mostly going to rely on some prior knowledge that you've acquired, but nevertheless, there is a bit of encoding that is happening. Encoding could happen in form of ontologies. We had just a track at this meeting earlier, and I see quite a few posters. It could be in form of mathematical models. In fact, some AI model, you could argue, is also, if it's doing well, maybe some form of knowledge, but just one that is sometimes hard to see through as human. So therefore, we have to think of data in a broad sense. I'm not just talking about the raw data when I'm talking about sharing. And so, again, I mean, I don't want to belabor this point, but we really have this data being produced in a specialized context, and then we need kind of to clean it, to organize it, to link it such that it can be then can be also understood without having to phone or texts or email the authors of the data to ask, what does this mean, or what sort of condition you've generated this data. Okay, and so what the funding agency and many people have put a lot of emphasis on is to ask researchers to contribute to this process, okay, to verify their data, to write DMPs, data management plans, and then revise the data management plans, and then tick all the boxes. So what do you do as a researcher? You finish your work, you're really happy, ready to submit the paper. Maybe you want spontaneously to share your data. Well done. Maybe you're being forced by the reviewers or by the journal. But that's also okay. So what can you do? So if there's some data that is kind of in a standard, well-known format for which there's some repositories, it's quite easy. You can just deposit this data. You put your code on, I don't know, GitHub, and deposit your sequencing reads into ENA. Okay, that's quite easy. Now, how about the non-standard data? Okay, that's where it gets a little bit more tricky, right? I mean, first of all, you may ask, okay, you may think, okay, well, then I can still deposit, say, my data into an institutional repository or into Zenodo. But then, in which format should I use? What ontology? Should I maybe come up with a new schema? We heard a talk about that earlier today. Well, that could take a while, actually, if we want to do a good job already understanding what exists already, and then, what sort of cross-references would be helpful. How fair is actually fair enough, in a way. And then, what would be then the value of what I deposit to people who are outside my field? How many people are going to then parse through my archive and then incorporate this data to then come up with new findings that can then be published in the scientific literature, given maybe the risk of confounders. Actually, we do not have that many success stories, despite all of the efforts that we've done into verifying data from researchers in that respect. And also, who's going to maintain this data? Once the moment you deposit it, databases starts to, your cross-reference starts to decay, genomes get updated, it gets quite difficult. And so actually we realize if we really want to do a good job, sharing non-standard data is kind of a project in itself. In fact, it is likely to be a longer project than the project that funded the work that generated the data in the first place. Because if you think about the life cycle of this data and the prospect about being reused, this will take a long time. So I'm not saying this, and also it requires somewhat distinct skills and motivation. I mean, we heard, and it's very encouraging, that quite a few of you feel that, that is also part of your objectives. But, it may be quite a parenthesis until your next discovery if you have to start to create a new ontology, and that could be, could be a long parenthesis. So this is, of course, not to discourage people from doing this type of data sharing of non-standard data, but we have to appreciate what this task entails. And we have actually some databases and knowledge bases and biodata resources that, the kind of the broader term, that is actually doing these efforts. The ones that have moved to that kind of third kind of activity that I mentioned at the introduction actually really contribute quite a bit to verification of the data, to data democratization. And I'm not going to go through the whole list here, but I do want to mention one function. I will get back to that in a few minutes, but that has to be recognized particularly in the case of curated databases and knowledge bases. It's one where you kind of aggregate information from multiple data or multiple papers and kind of synthesize some sort of consensus or some sort of a sense of what sort of knowledge can be extracted from that data. And if you think about it, this work is really at the heart of the scientific methods. is at the heart of that pyramid of knowledge that I mentioned, that is like trying to, go from maybe the primary literature to something that is kind of more of a, some takeaways and generalization that will contribute to knowledge. And so I would argue, it's not just enabling data sharing by, if you wish, just cleaning the data, but it's actually, in many cases, also really contributing to generating new knowledge. And then it is, of course, very unfortunate if that new knowledge that ends up in a repository, static repository that people cannot find later or is no longer maintained. And of course, it is also even worse if there's been some efforts to extract this data in a resource and the resource ultimately has to close down because of lack of funding. So that's an important message. Data resources are critical enablers of data reuse and fair. If you think about the success stories about data reuse, quite often you have a database behind it. So that unavoidably brings me to the topic of funding. We saw this coming a little bit, but I mean, it's most data resources suffer from chronic funding difficulties, and I did want to just mention like the talk of Amos Barak, who very sadly passed away in the last year. He was a founder of SIB. He created quite a few resources, including SwissProd, the NextProd, the Celosaurus. And I'm not going to belabor the point about the fact that data resources suffer from chronic lack of funding, but if you're interested in this topic, his talk from last summer in Liverpool is available on YouTube, and I really recommend. It's not that long, and it's really quite engaging. And it makes the point very well that it's not a new thing that databases are not getting much support. Usually, we see that the funding of databases and knowledge base is happening either, well, the first type of funding is like individual researchers who maybe are bootstrapping the effort from research money or their own kind of time, and in some cases, like still continue this as a hobby. And then really when it starts to be a bit more serious is when there's some project grants that are supporting these databases. But of course, project grants is problematic. The excellence criteria that are used for projects, they don't really apply very well. They don't translate very well to databases. And the gaps actually in funding are much more dramatic in the case of database and basic infrastructure because of all the dependencies. And then, okay, then if you have to let highly specialized people go, it would be difficult to recreate these skills. People may, you could see what happened already in the past few months with NCBI taxonomy. Just the mere existence of like a message that says, well, this database won't be updated over the weekend is already causing like the community to start questioning their reliance on the database. So stop and go is highly damaging. So some message we try to get across is that the status quo is actually very detrimental to funders' own goal. And so, in a way, to rely just on researchers alone to achieve FAIR is ineffective and wasteful. And so, actually, a little bit of help through the database would go a long way. And if you think also about, I'll get back to that in a few seconds. And so the remedy for that is quite simple. The funding agency should provide dedicated and also competitive. Of course, there's always a limited budget. But there should be some instruments to support data resources. Okay. And that would be better for science, and it would be also better for the very narrow goals of these funding agencies. So I want to give the example of how we do things at SIB. Provides some examples. So SIB, thanks already for the kind introduction. I would just mention it's structures as a foundation, but where we have agreements with all the major research institutions across Switzerland. And so through that, we have a network of about 90 affiliated research groups, about 900 members in total, but we also have about 200 employees of the foundation that are either embedded in these different sites or concentrated in a few central support function and internal groups, mostly on the infrastructure in Geneva, Lausanne, and a little bit in Basel. And indeed, we serve as Elixir Switzerland. And so our activities are three main parts. So one is this open database and software that I mentioned, and you will recognize a number of them. And I'll get back to that in a second. But there's also the Center of Excellence. This is the part which you can think it maybe mirrors a bit also some of the activities that are done by l'Institut Français de Bioinformatique with services and competences for academia, but also industry, the government. Typically and training also, and typically in areas that are connected also to our data resources. And then we have the coordination aspect. So the fact that we are an independent foundation already in Switzerland is quite helpful to be like sometimes the broker when different institutions that feel in competition with one another needs like a non-threatening partner to, for instance, take care of the data or some standards. But we also play sometimes this role also internationally in some collaboration or some efforts. And here's a number of projects, mostly around Switzerland here. And so I did want to mention the funding model for resources in Switzerland, which I think would be helpful. I mean, first, I think this has been in place for a number of years already, and it is stagnating. I'm getting already a little bit to the punchline in Switzerland, but I think it's not been adopted in many European countries, and it's a pity because I think it's been, in some sense, like quite a success story, this funding model. So the way it works is that all the resources are led by SIB group leaders that are within Switzerland. So anyone who's a member of SIB can every 4 years make a case that they think their resource deserves support at the national level. And then we will not cover it entirely. The resource then is developed in collaboration with SIB, but we can provide some more stable funding, for instance, to hire some engineers or some curators who will kind of, in a way, be outside of like the typical academic positions, and then that can help with the critical continuity of the research, of the resource. And that's about 7 million Swiss francs, so about 7 million euro a year that is invested in that manner from the government. That's not the full cost of these resources, but it's a really critical enabler. And so it's, it is substantial, but it's also not enormous if you think about the scale of a country and also, the leverage that this confers. Because then these resources are used really quite a bit. So these are the figures from last year, 27.5 million users. This is a very big growth also due to AI bots. It's hard sometimes to know who is a real user, but in any case, and apologize on behalf of my colleagues if you had some difficulties accessing some of our resources in the last year, because like many people, many resources, we suffer also from like now this big influx of use. About 27,000 citations last year to these resources, but also interestingly, they are also used quite a bit for innovation in patents. It's about 10,000 new patents every year that are citing SIB resources. Of course, it's used also by many AI methods. And so as a result of this long-term investment, I think it's fair to say that Switzerland has some recognition internationally for the continuity and the dependability of these data resources. But I did want to mention that funding has been flat for us for the last 10 years. I mean, we, again, don't want to complain about this, but it is a challenge, and it means we need also to think how to use this resource really to have like the most impact for every franc invested. And we think also by having kind of a more big, broader movement also around Europe and around the world into this investment in the resources, I think everyone's going to be better off. So I've argued now, I hopefully somewhat convinced some of you I'm sure there are still many skeptics, and in fact, there are obviously many skeptics because the funding issues are not yet solved. And you know that just in the last year, there's been some resources like POM-based that have had like their funding cuts and is causing some real concerns. So what sort of criticism do we hear? And then maybe what are some of the answers we could have? So one thing that I often hear is like, well, funding data resources is expensive. Okay, well, it is expensive, but doing research is expensive. We are all quite expensive. So we have to think not just, we have to think in relative terms. And there's been some studies that suggests actually that if we would invest just 1% of the funds that are allocated to project funding for the maintenance of resources, that we will go a long way addressing this issue. So there are these two papers that I recommend looking at. And of course, this is not even looking at the cost, of course, of not funding the resources and thinking about all the time that is wasted reinventing the wheel or recreating data that you might have been able to use to come to your conclusion, or even worse, like redistilling the knowledge that has been previously captured. So the criticism number 2, very common, will be to say, well, we recognize these databases are really important, but it's really not really our problem because we just fund bottom-up science, for instance. Okay, this is just, this is infrastructure. It's not part of our budget. So there, I think it's important that even if the funders are taking a very narrow view of their mission, I think we can make a very strong case that the databases are integral to that. I mentioned already my arguments regarding, FAIR, but actually, if you think fundamentally, and I want to go back to also the previous contribution I mentioned of the databases, it is really a critical enabler of new research. So quite often, the databases are the input to new research. As I mentioned, they contribute to generating new knowledge, so they're in the middle of the research, and at the end, they contribute to the spreading of the outputs. Okay, so actually they are at the beginning, at the middle, and at the end of the project. Actually, they have their relevance. And actually, interestingly, funders themselves increasingly recognize that maybe, you might remember DORA, the notion of the DORA agreement was that it's not just high-impact journals that are relevant, there are also some other journals that publish worthy science, so we shouldn't be just looking at impact factor. Now there's COARA, I don't know if some of you may have heard of this, but is a notion to say, well, it's not just the papers that are important for science, actually there are also some other roles. And so it's a bit ironic that the funders who claim that, through signing COARA and requiring narrative CVs so that you can explain all your other contributions, that they themselves then think that funding activities that are outside of like publishing high-impact papers is actually not really their remit. So it would be also consistent with their own kind of engagement. We see even sometimes claims that, that funding resources is to the detriment of new research, which I think is quite ridiculous. Okay, so third criticism, we cannot just keep on funding more and more resource. Okay, that's fair. It's, as I mentioned, is a long-term commitment. And so what is the solution to that? Well, the solution is that we need to make some tough choices, just like with bottom-up research. And that's what we have to do also at SIB with our limited budgets. And that works. And so you need to think about, where and when you can make a contribution. And so for us, we think it's when you have like an early stage resource, that should be funded through research fund and local funds, and only when it's really clear that the need is established does SIB step in, and then there will be some maturity, and we still have to make some tough choices, and at some point, it will move into legacy, so then we will stop funding it, but the PI, maybe they still find a way to get it survive in one way or another. So there's a rigorous competitive process to that. I'm really grateful that people like Claudine Médic sit on our scientific advisory board and help us sort through external peer reviews to make these difficult decisions. And I want to mention that so we had this flat budget. I say 10 years, actually it's 12 years. Time flies. And over the last 2 4-year cycles, actually, we had to make these choices. We added new resources, we sunsetted some, and in fact, I can say that we've even had to reduce the funding to some of the highest scoring resources just to make space for emerging needs. But we think this is a reasonable model. It's not one where we expect, like in a project, something new every time, but there is a limited budget and there is an objective, and the people who review this understand what it means to provide an important a resource. Okay, I'm almost through this criticism. So number 4, why should we pay if other countries use our resources as well? Okay, that's a very big problem, turns out. I mean, I took here the point of view of the government, but we had some funding crisis last year with, some cuts to NIH funding. And then when we talk to our government, they say, well, it's great that you have so much impact on industry. Why don't you ask them to pay for Swissprot because they file all these patents? And so then we talk to the industry and they say, we really love your resource, but, and we would like to help, but how can we help if our competitors don't have to pay? Okay, so why don't you ask our competitor first? And if others also agree, maybe, And so in a way, because it's helpful, it's valuable to many, people are just thinking, okay, someone else surely will pay for it. It's a real problem. But I think it's very interesting to see that actually being a shareholder into a resource, as a PI or as a scientist, to have a stake in a resource that is used will generate also some scientific credit, some recognition. You might get some new collaborations. You will feel that you're doing something worthwhile. And actually, you can be generous and at the same time also, in a way, benefit also disproportionately. And so this is the argument that we are trying to make, and I think there's some justification for that, because we see that funding biodata resources also provides some economic and strategic benefits. So what might these be? So one thing which we see now is being discussed quite a bit because of the geopolitical instability is how to secure your resource access to the data. If you're just relying on other people's funding the data, then it's going to be potentially difficult. And we can see data is becoming, it's the new oil, it's really important. And so, governments need to ensure access to this data, but also by funding it, they also have a lot of impact on how these resources are developing and also how they are aligned with their other priorities. And then there's also the skilled workforce argument. It's also very clear that if you make these investments in your institutes, in your country, then you will have also people who are quite competent about this data. And that also generates some positive externalities. That's also great if you want to attract then companies who will then go to places where you have a skilled workforce. And yeah, and so even completely open data can have a strategic value. I mean, with mobile phone, Google open sourced Androids when they needed to compete with iPhones. I mean, they open sourced their software. They are not exactly known for being a company that just wants to create the biggest positive externalities for the world, but openness can also confer some strategic advantage. And we think particularly in the area of data, that is the case, because if you put your data out there, others are also integrating your data, linking this data, it can become kind of a de facto standard, and a little bit like maybe the US dollar being kind of the world's currency is advantageous to the US. Maybe UniProt being kind of the de facto identifiers for protein is also advantageous to UniProt and to Switzerland. And also, of course, it's also quite a bit more efficient if we can keep things open rather than having to charge, hire some salesman and accountant, etc. And I can give as a little story that when I was a young PI in London, I was really struggling to get some collaboration with industry. And then once BASF, Bayer at that time, contacted us and asked if they could use some of the software that we had in the lab, which had a license that it's free for academia, but if you're a company, you have to ask permission. And because I thought, okay, that way I can at least justify this to our funders. And then, and so they asked, can we use it? I said, yeah, sure, that's fine, use it. And they said, oh, no, but we need to have a contract now. We need to, I cannot just tell my boss that I'm allowed to use it. And so I was starting to think, okay, so now we have to involve the university. Who's going to draft the agreement, etc. It's going to be really complicated. By the time this is finished, they will have lost interest. So then I said, no, I make it open source. That was very quick, and then they could start using it. And then in the end, this gave rise to some sort of collaboration. So that can also be a reason. And actually, another reason I've heard also that is quite selfish to open source is a colleague who was changing institution and was really concerned that they will lose access to their own data. And so they thought, okay, let's open source it. And so when I get to the other institution, I can still have access to it. So whatever the motive may be, we think, this is, it's interesting to see that there's still some good arguments to go into open data. But I think the most actually important argument is that funders of all people should understand why it makes sense to do these investments in your country, even if you do not get an immediate return on investment. Because it's actually the same reason. No funder is asking, for instance, that the papers are just limited to readers from their own country. Everyone understands that actually it is advantageous to be exporting these results and to attract the bright scientists to their country. And so why should it be any different with respect to databases? And yeah, we think there are some evidence that countries funding resources reap disproportionate benefits. And I'll just mention this one study of the value of UniProt, which was really quite an eye-opener. And actually, this one actually looks at the number of patents that are actually enabled by UniProt. And then there's the top 5 institutions worldwide. There are 3 that are Swiss companies. And actually, interestingly, one of I don't know exactly the rank, but among the top 5, 3 are Swiss companies. One is Inserm in France, and one is Novo Nordisk. But this was actually very interesting kind of evidence that it's also good for the local economy, even if everyone can use the data. So I just want to now recap the roles and responsibilities. So as researcher, I think, of course, it is our duty to deposit standard data according to field practice. And once in a blue moon, if we really find we sit on some data that we think really could be really useful, perhaps consider then developing a data resource. But it's a long-term commitment, and it will, I guarantee, it will distract you from doing your discovery-led science, but it may also be well worth it. And I will say also, yeah, it's a bit like adopting a puppy. It's all fun at the beginning, but it's a long-term commitment. And do collaborate. If you are not doing it, still collaborate with the data resource provider to make sure that your output is then used there. So for the data resources, we like to really also remind our resources to prioritize their efforts to the activities that maximize the democratization effect of the data. There's a lot of activities that you could be doing, but actually ultimately this is what is going to have the most impact. And collaborate with researchers. And so for funders, again, with an institution journals, requiring deposition of standard data, recognizing and rewarding data democratization, and then funding data resources through dedicated schemes. Now, I have one last slide before the conclusion. I'm so sorry. I thought I will just not have too many slides like this. We can have lots of time for discussions. Okay, for the early career researchers in this room, I think it's worth thinking about, kind of which role suits you best. I mean, you have still at a time where you can straddle, but they are actually very rewarding careers, I think, in all three categories that I mentioned. And, I can mention, so for instance, when I started the lab, I was very much focused on methods, and then I realized, well, actually, it would be good also to work on resources and then applications. So I kind of felt we need to hire people who have complementary roles. And so actually, in our lab, we really try to value both the methods, the resource, and application, and we see this as a virtuous circle. A new method will then be disseminated in the database, and that opens up the door for collaboration with application. And I want to recognize Adrian Altenhof, who's been kind of leading the resource side of the lab for many years, and Natasha Glover, who's really brilliant in terms of leading the application also. And actually, we co-lead the lab with Natasha these days. And, this is I think, applicable for many people, but actually there are some people who can do it all, the rare unicorn. So we had one guy in the lab a few years back, Yanis Never, who is now at the University of Strasbourg, who was equally good at doing all of these things. There will be some of you who are like this, but I think even you have only 24 hours in the day, and so you may still want to collaborate with people who have strength in the other areas. And if you are straddling multiple roles, I think it's really helpful to think about the role that you serve in a particular project. And a lot of the difficulties we face is when you think you should be a researcher on the project, but you're treated like a service provider, or on the contrary, you're a service provider, but you're not paid as one, and then you may end up like not being able to deliver on the expectation. And so actually I could elaborate on this, but given the time, I will stop here. And yeah, I mentioned that is rewarding, and that there are many interesting enabling roles as part of data infrastructure. In particular, I think France is really a fantastic place for that. And then you have with the society and the Institute of French, Institut Français de Bioinformatique, many really exciting activities, and I'm not surprised that so many of you are also identifying with this category. Okay, well, I'm not going to belabor the points. Thank you very much for your attention. Acknowledgement. So, did want to mention Amos. It's been very sad for us, his passing, but we're very grateful for all the work, and colleagues who've been exchanging a lot of these ideas also within Elixir. In the last year, there's been a number of papers, collaborative papers also on these topics. I invite you to have a look at them. And we have an event at the end of the summer. It's not too late to join if you are still on the fence. And also, again, for early career researchers, there's a European Student Council Symposium. You can register just to that. It's taking place on the Thursday if you like to join us in Geneva. Thank you very much for your attention.
So thank you, Christophe. It was enlightening. I mean, I'm already a groupie. I guess there are some questions. So who starts?
Thank you for your very interesting presentation. I love the fact that we spent 10 more money in publication than in infrastructure. I would like to have your idea about the DSI and the sovereignty of data for non-European countries, let's say, especially southern countries.
What is DSI?
Digital Sequence Identifier. All the sequence that can be under the framework of the Nagoya system.
Yeah. So there are these efforts to So Nagoya, for those of you who are not so familiar, that was about trying to limit biopiracy, so the notion that developed countries would just go and help themselves in developing countries and then sell this back as medicines or valuable compounds. And then there's the realization now that, well, actually sequences sometimes can be also quite valuable, and they can readily cross borders in a way that is much more immediate, and so there should be some benefit sharing, and so there are some discussions happening. There's some resolution at the COP15 conference where there's a so-called Cali fund that is going to be established to try to require contributions by companies that benefit from data that are coming from the Global South. And there's similar conversations that are happening at the WHO, also specifically about pathogen data. And so I think it's a very interesting development. Obviously, this is negotiated by diplomats in many cases who don't understand very well the challenges associated with data, and also the notion also of benefit can be quite different depending on who you're talking to. For researchers, it's often the notion of being on the paper or still having an intellectual involvement in the work that is derived from this data. Obviously, there's also in places where it's more the economic value that is derived, and so it's quite a complex discussion. But I do think that this is affecting the stance we've had on open science, because it means, in effect, that we cannot, that there's some duties, some moral duties, and more than moral duties, actually, there are some international treaties for some users of this data to then pay in some fund. And so as academic database, we do not need to pay according to these points, but we need to make our users aware. So this means we need to provide information about the provenance of this data, and we have to add some restrictions. And of course, we see with the data spaces and this notion about sovereignty, there is a push towards kind of more walled gardens. And it is worrisome because you sometimes, the remedy can be worse than the problem in the first place. Openness also can have a lot of advantages. I'll try to argue in my talk also about some of the strategic advantages. I'm not sure, for instance, that being credited in the supplementary material as one of a million other authors by depositing your data in Gized is a great academic reward for our researchers who would have been reluctant to deposit their data into INSDC. So it's a complex discussion, but I think we have to have it with some humility. Yeah, thanks.
Okay.
Hi. So thanks a lot again for the fantastic work you're doing in Switzerland. I will join my colleagues to say, yes, it's very valuable and for a long time, so thank you. One thing about the reliability of your tools is the fact that all of the data are of very high quality. And part of that is because you have human experts validating them, and it is seen on the website. It is explained. With the availability of massive machine learning techniques, one way to change our vision of the knowledge is to get one of these tools for scientists, by scientists. And I think maybe you are going into this direction, but there are, of course, the problem of the sources and of the quality. So what do you think about that?
Yeah, I've not gone into this topic because it could also be quite lengthy. I will be just brief in the interest also of having some other questions, but I think the expert is, we cannot have one size fits all, and the detail really matter. And if we want to be able to push and use these tools and develop tools that are effective, there will be no shortcuts. We already see in some of our teams that it can be used to accelerate the process, but you absolutely need to be critical of how it works, and the importance of benchmarking is going to become even more important. In fact, it's a bit of a shame that even before AI, benchmarking was often treated a bit as a kind of an afterthought, but I think to the extent that we've ignored it or minimized it in comparison to, let's say, creating new elegant algorithm, now with AI, it's like, it's not, the AI method is not so elegant. And so the only thing that we have is perhaps some reasonable performance in benchmarking. And for that, again, you need the experts because, I mean, we all know meaningless benchmarking results that were obtained by misunderstanding the data. And so for that, again, I don't think there's any shortcuts. Yeah.
Okay, next question.
Hello. At the back on your right here. Thank you for your talk. You said you had more to say about early career researchers when they can choose between being provider or researcher. And I was wondering how an early career researcher could go and focus on the resources when they are in France and not in Switzerland. Is there any institution or anything that is close to the SIB or that you can think of?
So, of course, you're very welcome to come to Switzerland. But actually, there's really a lot of fantastic work also happening in France. I really want to acknowledge that. So I mentioned all the collaboration we do, in particular with Elixir France and the Institut Français Bioinformatique at many different levels. There's many people who devote their career to support infrastructure. But actually, I mean, the first time I was in Strasbourg was to meet Odile Lecomte. Where is Odile? Is she in the room? And Julie Thompson. Salut Odile. And Julie Thompson, you know, Julie has been working on the Bali-based resource and Clustal, and there was Ortho Inspector, and there are many resources. I mean, I have also, for many conferences, I was bumping into the folks working on Greenfield, the Greenfield database that is looking at plants from the Global South. And so actually there's tons of activities. It's true that it's hard to sustain this funding. This is really a problem. But I have to say, I think I'm quite optimistic that the importance is becoming so overwhelming that actually there are some responses. And to give an example, I mean, we really had this crisis with Uniprot last summer, a couple of months that were very, very difficult, where there were dozens of curators actually and people at risk. And so I was really very surprised by the response still. I mean, and you know, we in the end had a bridge from Novo Nordisk, and now the European Union has like a dedicated scheme that they are going to have this autumn specifically to support this resource. And so I think, I mean, sometimes, especially at the beginning of your career, you're very worried about what is expected from me, how will I be judged? And I mean, you know, as long as you don't, I mean, you also need to have the next job. This I understand, and it's very easy for me to say, oh, you know, just believe in what's right. But I do think that if you're enabling others to do really good science and you stop, then people notice. And in a way, that one option was also to do a strike, which I think you have also some experience in France. And to do a strike could be a good way also to raise awareness. And so I do think that even if you do not see like a golden path to funding, there's still lots of ways that are possible in France. And the evidence is already just in the room and in the conference. Yeah.
So on these words, we need to wrap up. I'm sure there were other questions, so I encourage you to directly go and talk to Christophe. Christophe will be here until the end of the conference, so don't hesitate. So thank you again. So I have two small announcements, but important. So for those who plan to do the boat tour, so Botorama, it's the river cruise activity. The meeting point has changed. It will be on Quai du Sable, Ponton 1, and it's really close from the initial point, which was, it's 200 meters from there. So, Quai au Sable, Ponton 1 for the boat. And for those who were more on the European let's say, a vibe. Lieu d'Europe exhibition and visit of European districts. You have to meet at 5:10, so 5:10, in front of the JOBIM banner outside, and to be guided by a volunteer, or you can directly go there if you prefer. So this is it with the announcement, and I urge you to stay in the amphitheater, because especially if you want to know how France ecosystem is working, it's starting right now with SFBI and with all the networks afterwards.
Okay. Non. Hein? Oui. On commence maintenant? Ouais, je vous ai vu disparaître, je me suis dit, ils sont pas là. Bon, alors le temps que tout le monde se maille. Dans le programme, on avait un délai ou on commence tout de suite? Parce qu'il y a des gens qui pensent que c'est la pause là. Ouais. Qu'est-ce que... fait des trucs bizarres. C'est la mascotte, je sais, je l'ai vu tout à l'heure. Là. C'est lui, c'est lui. On est bon? Ou on attend encore? Ça se dissipe là. C'est pas grave. Allez, on fait la présentation dehors. Allez. Alors, bonjour à tous. Donc, je vous propose de commencer maintenant l'Assemblée générale de la Société française de bioinformatique. Alors déjà, merci à vous d'être restés. On va essayer de faire au plus vite. Donc on va avoir une première heure, une première session. On va vous parler de la SFBI, puis du groupement Mérite. Et ensuite, on aura une petite coffee break. Et on reprendra avec la présentation des réseaux et des groupes de bioinformatique en France. Donc, les questions généralement que tout le monde se pose, c'est qu'est-ce que la SFBI? J'ai essayé d'y répondre en début de la séance des JOBIM, mais bon, il y a encore potentiellement peut-être quelques questions. Comment nous sommes organisés? Quel est notre réseau? Et quelles sont les principales actions? Donc, alors, qu'est-ce que la SFBI? Comme je l'ai déjà dit durant le speech d'ouverture, c'est une société savante. Mais alors, ce qu'il faut savoir, ça c'est pour le petit historique, donc le premier JOBIM a eu lieu en l'an 2000. Il ne s'appelait pas encore JOBIM. C'était simplement un regroupement, une volonté de plusieurs bioinformaticiens à se retrouver et échanger ensemble. Donc, à l'issue de cette première rencontre, en fait, déjà, cette première rencontre a été un succès. Et les scientifiques ont souhaité réitérer cet événement et continuer à se retrouver tous ensemble. De là, ils ont eu l'idée de créer une société savante. Et donc, comme on sait que les démarches administratives en France sont très rapides, c'est en 2005 que la SFBI a été créée. Et plus précisément, le 1er juin. Donc, merci à Alain Guénoche pour ça. Et du coup, on a commencé à appeler cette conférence JOBIM, en référence à Carlos Jobim. Merci à Marie-France Sagou pour cette inspiration. À l'heure actuelle, donc, la Société française de bioinformatique est une association loi 1901. Donc, nous n'avons pas à viser de faire des profits. Et nous sommes détachés de toute institution. C'est-à-dire que nous ne sommes pas ni CNRS, ni INSERM, ni universitaires, ni INRAE, ni INRAE, peu importe. D'accord ? Donc nous sommes, on va dire, représentants de la communauté et nous pouvons représenter la communauté et porter très haut la voix de la communauté, c'est-à-dire votre voix. L'idée, c'est de promouvoir la bioinformatique et surtout l'interdisciplinarité. Donc, ça veut dire aussi bien la biologie moléculaire, l'informatique, les maths, les stats, mais aussi la physique et d'autres disciplines qui recouvrent notre domaine. Et l'idée, c'est de réunir tous les bioinformaticiens ensemble durant cette conférence annuelle de bioinformatique. Et de discuter ensemble de tout ce qui est formation, intégration des jeunes bioinformaticiens, surtout ce qui a été un point important des dernières décisions de la SFBI, et encore aujourd'hui. Alors, que sait-on sur la SFBI en 2025? Donc, pour information, point de vue santé financière, ça va. Voilà. Vous aurez les informations sur la slide plus tard. Le bureau de la SFBI est actuellement localisé à Montpellier depuis octobre 2021, là où je réside, puisque je suis la présidente. Nous avons donc des adhésions. Donc, j'espère que pour la plupart d'entre vous, vous êtes adhérents à la SFBI. Nous, on fait en sorte, par rapport à beaucoup de sociétés savantes, je vous encourage à aller regarder et à aller vous renseigner, à garder des frais d'adhésion vraiment dérisoires. Donc, pour les jeunes non permanents, c'est de l'ordre de 10 €. Pour les permanents, c'est de l'ordre de 20 €. Nous avons également, alors en 2025, on a comptabilisé 447 adhésions individuelles. Donc, merci à tous nos adhérents. Ce qu'on peut voir juste sur le plot en bas, sur le graphe, vous avez le nombre d'adhésions par an. On voit que ce nombre d'adhérents est quand même assez stable. Donc, on est aux alentours de 400 à 450 adhérents par an. Nous avons également des adhésions morales, c'est-à-dire que si vous souhaitez que votre labo, votre équipe soutienne la SFBI, alors généralement on vous envoie un petit mail en fin d'année lorsque vous avez des restes de fonds. J'espère que vous en aurez encore, vu la situation actuelle. Et donc en adhérant, en cotisant pour une adhésion morale qui s'élève à 300 €, vous permettez à votre laboratoire de devenir adhérent, ou à votre équipe adhérent de la SFBI. Depuis 2022, nous avons à peu près 20 adhésions par an. Donc en 2025, ça se levait alors de 25. Vous avez sur la carte plus bas les différentes adhésions morales. Donc on voit que les adhésions recouvrent bien toute la France actuellement. Et ici, je vous présente simplement les logos et je tiens à remercier tous nos adhérents moraux, tous ceux qui supportent la SFBI, aussi bien ceux qui réitèrent leur soutien d'année en année que ceux qui décident de nous soutenir ponctuellement. Alors, qu'est-ce qu'on fait avec ces adhésions concrètement? L'idée pour une société savante, c'est de ne pas faire de profit, c'est de vous redonner ces adhésions. Alors, comment on vous redonne ces adhésions? Première chose, c'est qu'on met en place des discounts, donc des tarifs préférentiels pour l'inscription à la conférence JOBIM. Donc, j'espère que vous en avez tous profité pour adhérer. Vous êtes également, à partir du moment où vous êtes membre de la SFBI, vous êtes également éligible pour postuler au prix de la SFBI, mais également aux bourses de voyage de la SFBI. Si vous souhaitez éventuellement organiser une conférence, une animation, un événement, que ce soit de la vulgarisation ou un événement scientifique, là, vous pouvez également faire des demandes de financement à la SFBI. Et depuis quelques années, on a également mis en place des gardes d'enfants, parce qu'on est bien conscient que la période des JOBIM tombe un petit peu avec la fin d'année scolaire. Et pour ceux qui ont des enfants en bas âge, si vous souhaitez, vous avez besoin d'un soutien pour faire garder vos enfants, ou même venir avec vos enfants à la conférence et les faire garder sur place, vous pouvez faire une demande d'aide à la SFBI. Si vous avez d'autres idées, nous sommes également preneurs, puisque l'idée, c'est vraiment de soutenir l'entièreté de la communauté. Un autre aspect bénéfique de la communauté qui est non négligeable et sur lequel on tient à insister, c'est que je pense que vous utilisez tous peut-être la liste bioinfo.fr, bioinfo@sfbi.fr. Et ce qu'il faut savoir, c'est que cette liste regroupe plus de 5000 personnes, donc 5000 scientifiques qui sont à la fois sur le continent, mais également partout dans le monde. Donc on a là une communauté qui est très très large et très très dynamique. Donc n'hésitez pas à utiliser aussi cette mailing list pour communiquer avec l'entièreté de la communauté. Alors comment on s'organise à la SFBI? Donc on a un bureau, comme beaucoup d'associations. Donc un bureau composé de 9 personnes. Ces 9 personnes sont élues pour des mandats de 3 ans, pour 2 mandats de 3 ans. Et au sein du bureau, on décide par la suite, on élit le président, le ou les vice-présidents, un secrétaire, un trésorier et des councilors. Nous avons également mis en place depuis quelques années le Conseil des sages, c'est-à-dire qu'afin de garder l'historique de la SFBI et surtout de savoir ce qui a marché, ce qui n'a pas marché, ça arrive, mais on décide tous les ans de demander aux anciens membres du bureau de la SFBI de faire partie d'un conseil. On peut être amené à les consulter, donc à faire des réunions avec eux 1 à 3 fois par an, de manière à avoir leur opinion, par exemple, sur des sujets sensibles, des sujets où on aimerait se positionner, par exemple. On a également, depuis 2023, des volontaires. Donc, je les remercie beaucoup. Et vous pouvez tous être volontaires. Si vous souhaitez aider la CFBI sur des postes et des missions bien précises, où vous avez des idées, n'hésitez pas à venir nous voir. Au stand de la SFBI, vous verrez une petite boîte en bois. C'est une boîte à idées. Donc, si vous avez des idées, n'hésitez pas à les mettre dans cette boîte également. Donc ici, je vous présente le bureau de la SFBI de 2025 à 2026, donc qui est composé, comme vous pouvez le voir, de scientifiques, donc de bioinformaticiens qui viennent un petit peu de partout en France et avec différents backgrounds aussi. Donc Audrey Bioué, Elodie Darbo, Sandra de Rosier, Charles Cellier, Cyril Noël, Delphine Potier, Pierre Poulain et Marie-France Sagot. À l'heure actuelle, je suis donc présidente de la Société française de bioinformatique et j'ai comme vice-présidente Elodie Darbo, Sandra de Rosier et comme trésorier Cyril Noël. Alors comment on s'organise? On ne peut pas tout faire. Donc on a décidé de s'organiser en groupe de travail. Bien évidemment, pour tout ce qui touche les adhésions, c'est Cyril Noël, notre trésorier, qui s'en occupe. Mais pour l'aspect communication, on va être plusieurs. La personne principale qui s'en occupe, c'est Sandra de Rosier, donc merci à elle pour ce gros travail. Sur le site web, on a aussi Sandra et Julien Fumet, un des volontaires, ancien président de la SFBI. Pour les bioinformations, c'est Elodie Darbo et Sandra. Et ensuite, en termes d'interaction avec les autres groupements bioinformatiques, on va avoir donc au niveau national Audrey Bioué, Sandra, Delphine Potier, Pierre Poulain, Marie-France Sagot et moi-même. Au niveau international, puisque ça c'est quelque chose qu'on a mis en place depuis peu, c'est Sandra, Charles Cellier, Marie-France Sagot et moi-même. Au centre, si je reviens sur les interactions, sur les formations, que ce soit les REBIF, donc ce sont des réunions qui ont eu lieu sur les formations diplômantes en bioinformatique en France, qui permettent de lister ces formations et de vous les mettre à disposition sur les sites de la SFBI, mais également réflexion sur les métiers, les MEDBIF. C'est Sandra de Rosier, Delphine Potier, Pierre Poulain, Marie-France Sagot et moi-même. Sur les bourses de voyage, lorsque vous envoyez vos applications et pour demander des financements, c'est derrière ces applications, il y a Elodie Darbo, Sandra et Cyril Noël. Pour les JOBIM, puisqu'on accompagne tous les organisateurs tous les ans, là également c'est Audrey, Sandra et Elodie. Pour les awards, c'est Delphine, Charles Cellier, Julien Fumet et moi-même. Et pour les interactions avec la COSAF, donc je vous détaillerai juste après, c'est Elodie Darbo et moi-même. Et bien évidemment, pour la mailing list, je finirai par ça pour remercier les volontaires, donc les bénévoles qui œuvrent dans l'ombre depuis de nombreuses années maintenant. Donc il y a Julien Fumet, Alexandra Louis, Laurent Mouchard. Et sur la liste aussi des emplois, Elodie Darbo, Cyril Noël et Delphine Potier et Alexandra. Je suis sûre que vous avez tout retenu là. Alors, les principales actions. Donc là, je vais passer la parole à Sandra, qui va vous parler des actions qu'on fait habituellement au sein de la SFBI.
Donc on voulait vous faire un petit bilan de tout ce qui s'est passé sur l'année écoulée impliquant la SFBI. Donc chaque année, on vous fait un petit bilan qu'on vous communique en début d'année sur le bilan des nombres d'offres d'emploi et de stages qui sont diffusés sur le site de la SFBI. Donc là, ce sont les chiffres pour l'année écoulée. Donc on a eu à peu près 700 offres d'emploi qui ont été postées sur le site. Donc vous pouvez poster une offre sur le site du moment où vous avez un compte sur le site web de la SFBI. Donc c'est ouvert à tout le monde. C'est très simple, vous avez juste un formulaire à remplir. Donc on vous encourage fortement à passer par ce canal pour diffuser vos offres. Donc aussi bien les offres de formation, donc ce que vous voyez à gauche, donc on voit qu'il y a une majorité d'offres qui concernent les stages de M2, mais également les thèses et d'autres stages de type Master 1 et également des contrats d'apprentissage. Et sur la partie de droite, donc vous voyez le nombre d'offres d'emploi, donc avec une majorité pour les ingénieurs d'études, mais également les postdocs, les ingénieurs de recherche, les autres ingénieurs, donc plutôt dans le secteur privé, et plein d'autres catégories. Donc ce bilan est accessible sur notre site web et vous avez également accès à l'ensemble des bilans sur les années passées. On vous propose également une représentation sous forme de nuage de mots où en fait on reprend les mots-clés qui sont utilisés pour taguer les différentes offres et on a pour habitude donc, de voir le terme génomique qui ressort assez fortement, mais on a également d'autres mots qui ressortent de plus en plus sur ces dernières années, donc avec le machine learning, le deep learning, l'intelligence artificielle, le single cell, les multi-omiques, etc. Dans ce bilan, vous pouvez également voir la localisation des différentes offres. Donc on voit que la majorité des offres concernent la France, mais pas que. On a également des offres un petit peu partout dans le monde, donc avec une grosse proportion également en Europe, mais également au Canada et aux États-Unis. Cette année, en faisant le bilan, donc avec Elodie, on s'est aperçu d'une petite évolution sur ces dernières années, et notamment une chute du nombre d'offres. Alors pas tant sur les formations, où on voit que le nombre d'offres de M2 d'apprentissage reste à peu près stable, mais par contre une diminution du nombre d'offres, notamment pour les postdocs et les ingénieurs. Donc on a choisi de vous montrer ces chiffres qui sont pas très positifs, mais bon, on voulait quand même vous les montrer et vous avez peut-être vu passer aussi une enquête. On a essayé de mettre en place une petite enquête donc, qui était assez courte, avec très peu de questions, mais pour essayer de comprendre un petit peu pourquoi cette diminution. Donc, est-ce que ça reflétait vraiment le marché actuel ou est-ce que les offres passaient par un autre canal, donc pas forcément par le site de la SFBI, mais peut-être qu'il y a d'autres canaux qui ont émergé ces dernières années et qui pourraient expliquer cette diminution. On essaye d'être un peu naïf, mais bon, voilà. Bon, on n'a pas eu énormément de retours de cette enquête, donc on n'a pas forcément de bilan à vous faire de cette enquête et de choses à en retirer. Elle est encore accessible, donc si vous retrouvez le mail qu'on a communiqué, n'hésitez pas à aller la remplir et à nous donner votre avis. On voulait également vous faire un petit point sur une action que propose la SFBI de manière récurrente, c'est les bourses. Donc la SFBI propose des bourses de voyage et également des bourses pour l'organisation d'événements. Donc comme pour la partie précédente, on fait un bilan chaque année. Donc on vous l'expose aujourd'hui et elle est également, ce bilan est accessible également sur notre site web. Donc au niveau des bourses de voyage, en 2025, il y a 24 participants donc, qui ont pu bénéficier de ces bourses. Parmi ces 24, il y en a 13 qui étaient allouées donc, à JOBIM et une pour la garde d'enfants. Donc Anna-Sophie, on a parlé juste avant. Il est vrai qu'on n'a pas beaucoup de demandes sur ce poste, donc n'hésitez pas, en tout cas si vous êtes dans ce cas et que vous avez besoin d'aide sur ce volet, n'hésitez pas à postuler. Donc ça représente un peu plus de 12 000 € de bourse sur l'année 2025. Et on voit en fait que nos jeunes bioinformaticiens ont beaucoup voyagé en 2025, donc en France, mais pas que. On a pas mal de conférences en Europe qui ont été couvertes et également en Asie. Au niveau des bourses d'organisation d'événements, donc on a des événements récurrents qui sont financés par la SFBI, donc notamment les Omic Days, le réseau local biotique, l'événement de GBIF qui est organisé aussi en marge de JOBIM et plein d'autres. Et donc ça représente en 2025 5 événements pour à peu près 2 000 €. Donc les bourses sont ouvertes toute l'année, donc n'hésitez pas à soumettre vos demandes tout au long de l'année. Le pôle de gestion des bourses étudiera vos demandes au fil de l'eau. Une des grosses actions dans lesquelles est impliquée la SFBI, c'est l'organisation des journées auxquelles nous participons toutes et tous aujourd'hui, donc JOBIM, donc la SFBI est le sponsor de JOBIM et fait partie donc du comité, du steering committee pour cet événement annuel pour la communauté. Donc qui est dédié donc à la communauté bioinformatique dans sa totalité, qui est plutôt à destination, enfin en tout cas où on essaye de mettre à l'honneur les jeunes scientifiques, mais pas que, pour pouvoir en fait présenter leurs travaux dans une ambiance un petit peu conviviale. En tout cas, c'est ce qui est mis en avant dans le cadre de ces JOBIM. Depuis quelques temps, on y reviendra juste après, il y a une collaboration donc, avec PCI pour pouvoir proposer des soumissions en collaboration donc, avec PCI. Et on a également donc, des prix qui sont proposés dans le cadre de JOBIM par la SFBI. Donc JOBIM est supporté par la SFBI et l'idée c'est de garder un petit peu l'esprit de JOBIM au travers toutes les éditions de chaque année. Il y a donc 2 partenaires, 2 sponsors réguliers donc des JOBIM qui sont le GDR-BIM et l'IFB, qu'on remercie grandement pour leur subvention et leur participation. Que vous dire d'autre? La CBI est impliquée donc, dans le comité de programme de JOBIM. Comme on vous l'a dit donc, en introduction, l'organisation de JOBIM est faite par un comité local, en fait, un comité d'organisation local, qui peut proposer donc, une coloration un peu différente chaque année, et puis quelques petites adaptations sur le format et le contenu. Donc notamment, il y a eu la proposition de mettre en place des mini-symposiums, aussi une session dédiée donc, aux activités de services et de plateformes. Ensuite, une année, un peu par la force des choses, une édition virtuelle et en tout cas hybride. Voilà, donc c'est des évolutions, des adaptations qui sont proposées chaque année par les différents comités d'organisation en local. Pour faciliter tout ça, en tout cas pour essayer de faciliter l'organisation de JOBIM, il y a un starting kit, un JOBIM pack en fait, qui a été mis en place où sont transmis chaque année au comité d'organisation et de programme différentes informations et différents documents pour pouvoir faciliter l'organisation, en tout cas aider à l'organisation de ces JOBIM. Donc il y a la charte, il y a une charte qui a été rédigée, qui est revue de manière régulière pour bien coller à l'organisation et aux différentes évolutions, un calendrier pour bien suivre les différentes étapes et ne rien oublier, des autorisations de diffusion, des templates pour les conventions, également pour la recherche de sponsors, le logo aussi qui est mis à disposition et adapté chaque année en fonction des différentes éditions. Donc JOBIM, ça change de lieu chaque année. Donc là, vous avez un panel des villes qui ont déjà pu accueillir la conférence JOBIM. Donc l'année dernière, nous étions à Bordeaux, l'année d'avant à Toulouse, et cette année donc à Strasbourg. Donc juste pour revenir sur l'édition de l'année dernière, donc l'édition 2025 qui a eu lieu à Bordeaux, donc c'était 500 participants en présentiel et un petit peu moins de 100 participants en distanciel. Donc avec un comité d'organisation, donc avec les 3 personnes qui sont présentes sur la slide, et puis également un comité de programme, un comité scientifique avec les 3 personnes citées, et un site web bien entendu qui est mis en place chaque année et qui reste accessible les années suivantes. Depuis 2022, donc, il y a eu la possibilité donc, de faire une soumission jointe à PCI. Donc ça, c'est quelque chose qui n'existait pas les années d'avant et qui a été proposé il y a maintenant 3 ans. Donc il y a eu un petit temps d'adaptation, on va dire. La première année, il n'y a pas eu beaucoup de soumissions. Là, l'année dernière et cette année, il y en a eu un petit peu plus. N'hésitez pas, en tout cas, c'est une possibilité qui vous est offerte depuis quelques éditions, donc n'hésitez pas à aller jeter un œil et puis à proposer des choses pour l'édition l'année prochaine. Comme je vous l'ai dit tout à l'heure, on a également la remise de prix lors des JOBIM. Donc l'année dernière, on a remis un prix pour la meilleure communication orale, donc à Juliette Audemars. qui était donc de l'Université de Bordeaux. Et on a également remis 2 prix pour le meilleur poster, donc à Hugo Bellavoir et Fiona Hack. Donc comme je vous l'ai dit, et comme vous le savez tous, nous sommes à Strasbourg pour cette édition 2026. Ensuite, on voulait vous faire un petit point aussi sur les réseaux de la SFBI, même si Anna-Sophie vous en a déjà parlé lors du discours d'ouverture. La communauté bioinformatique en France est très riche. Il y a plusieurs réseaux qui existent et on n'est pas chacun de notre côté, en fait. On communique tous ensemble pour animer la communauté et on a chacun nos spécificités. L'idée étant que la SFBI est un peu au cœur de tout ça et essaye de communiquer un peu avec toutes les parties prenantes. Donc sur la partie recherche et infrastructure, vous avez donc le JOBIM et également l'IFB, donc qui vont vous faire des présentations dans la session juste après. Sur la partie représentation, donc il y a la COSAF, donc on en parlera aussi juste après. Sur le volet santé, il y a également le réseau donc BioInfoDiag et également France Médecine Génomique. Sur le parti métier, donc vous avez le réseau métier mérite qui fera une présentation juste après. Et puis on a également une animation de la communauté des jeunes bioinformaticiens avec JBIF. Donc sur le site de la SFBI, vous pouvez retrouver le listing de tous ces réseaux avec quelques informations et surtout chacun des sites respectifs pour pouvoir aller retrouver l'information plus facilement. Donc n'hésitez pas à nous faire de retour d'ailleurs si des fois vous voyez des informations qui seraient d'utilité et qu'il faudrait rajouter sur notre site web à ce sujet-là. La COSAF. Donc, ce qu'on voulait vous dire, c'est que la SFBI est un membre actif du Collège des sociétés académiques françaises, qui a été créé en 2021. Voilà. Donc, et on a pris part en fait à plusieurs actions de la COSAF, donc qui sont listées ici. Donc ça, c'est les dernières actions pour lesquelles on a pris part. Donc participation à des événements, prise de position aussi sur certains aspects. On a également co-signé des tribunes. Et puis on a également, donc on vous a mis un événement donc, qui est en cours en ce moment. Donc n'hésitez pas à aller jeter un petit coup d'œil. Donc là, on n'a pas mis de lien, mais en tout cas, on avait diffusé l'info sur la liste bio-info de la SFBI. Donc vous pouvez également vous positionner. Donc n'hésitez pas à aller regarder un peu les différentes actions. On essaye de vous communiquer, en tout cas, de vous diffuser toutes les informations qui sont d'utilité pour la communauté. Donc là, c'était plutôt le réseau, les collaborations avec les différents groupes en France, mais on a également un réseau à l'international qui a quand même bien évolué ces dernières années. Et on en est assez fiers, comme l'avait dit Anna-Sophie lors du discours d'ouverture. Donc on est membre affilié de l'ISCB depuis déjà quelques années. On a également des rapprochements donc, avec différentes sociétés ou différents réseaux à l'international, donc notamment la société brésilienne et également le réseau d'Afrique de l'Ouest. Donc je parle de cela en priorité, enfin en premier on va dire, parce qu'on organise un mini-symposium donc demain après-midi qui met à l'honneur ces deux communautés. Donc n'hésitez pas à y participer si vous êtes inscrit en tout cas. On a également des rapprochements avec la société d'Amérique latine et également la société espagnole et allemande et italienne. Et vous verrez à la fin de la présentation qu'il y en a d'autres. On voulait vous faire un petit focus sur notre participation donc, au réseau Rabias, donc le réseau en Afrique de l'Ouest, où on a eu la chance de participer et d'être invité donc, en fin d'année 2025, au mois de décembre. Donc, on a pu participer donc, avec une délégation de l'IFB, de la SFBI et de l'Université de Montpellier. Donc, on a participé donc, au lancement du réseau dédié aux bioinformaticiens et aux administrateurs systèmes d'Afrique de l'Ouest. qui était organisé donc, par Wave et puis par l'IRD. Donc on a la chance qu'ils soient venus donc, participer à l'édition de JOBIM cette année. Donc on en profite pour les remercier grandement de cet événement et de ces échanges qui ont été vraiment très très riches. On voulait également, donc on avait communiqué un petit peu l'information et on en profite parce que Sophie est parmi nous. On voulait la féliciter puisque Sophie a été en fait élue ISCB Philo, donc l'édition de 2026. Donc un grand bravo à Sophie pour cette reconnaissance et tout le travail que Sophie a fait pour la communauté en France. Et le dernier volet, donc le dernier point qu'on voulait aussi vous partager, c'est le travail qui est fait donc, au niveau de la communication et des réseaux. Donc, comme on vous l'a dit, il y a énormément d'informations qui passent donc, par notre site web. Il y a beaucoup d'informations, en tout cas on essaye de vous partager beaucoup d'informations. On essaye aussi de le rendre accessible au plus grand nombre, donc c'est pour ça que ces dernières années on a essayé donc, de traduire une bonne partie du site web en anglais. Donc, il n'est pas encore totalement traduit en anglais, mais en tout cas le contenu principal est accessible donc, en français et en anglais. On a également mis en place ces dernières années des annuaires. Donc, on vous avait présenté l'annuaire des membres de la SFBI l'année dernière qui était assez frais. Donc il devait y avoir une vingtaine de membres, donc qui ne reflétaient absolument pas le nombre de membres de la communauté. On a un peu augmenté, on est presque à 100, mais bon, on est bien plus que ça, donc on vous encourage vivement à aller activer sur votre compte cette fonctionnalité. Et puis on a également un annuaire des équipes en bioinformatique, donc qui regroupe à peu près 400 unités ou laboratoires. Donc pour vous ajouter à l'annuaire des membres de la communauté, il vous suffit d'aller simplement sur votre compte, sur le site web et de cocher ces petites cases. Donc ça vous prend vraiment 30 secondes et ça vous permet donc d'être visible au sein de la communauté. Donc outre ce site web donc, qui contient beaucoup d'informations, on a également d'autres moyens de communication, donc la fameuse liste de diffusion que vous connaissez à mon avis toutes et tous, donc bioinfo@sfbi.fr, qui est grandement utilisé par la communauté et on vous encourage vraiment vivement à l'utiliser pour diffuser le plus largement possible vos actions et vos événements. On est également présent sur les réseaux sociaux, donc sur LinkedIn, Blue Sky et Mastodon. On a également une newsletter donc, qui s'appelle les bioinformations. Donc il n'y a pas que la SFBI qui rédige cette newsletter, c'est vraiment tous les groupements en bioinformatique, donc le GDRBIM, l'IFB, les GBIF, qui contribuent à rédiger des articles. Donc c'est une édition par trimestre pour vous transmettre toutes les nouveautés, les actualités, les informations, pour que vous restiez au courant de tout ce qui se passe. Et vous avez également le blog de bioinfo.fr et le Discord qui contient aussi plein d'informations extrêmement utiles. Et je vais repasser la main à Anna pour le bilan financier et moral de la SFBI.
Merci Sandra. Alors, je vous ai dit que la SFBI était en très bonne santé financière. Hop. Ce qu'il faut savoir quand même, c'est que voilà, donc au 1er janvier 2025, sur notre compte, on avait 103 000 €. Donc tout allait très très bien. On a plusieurs recettes. Donc dans les rentrées d'argent, on a les adhésions SFBI qui représentaient 5 900 € en 2025. On a les clôtures des JOBIM. Donc tous les ans, généralement, le JOBIM actuel, par exemple, là, on n'a pas encore clôturé le JOBIM 2025, donc il nous manque les recettes du JOBIM 2025. On a des frais aussi de gestion de compte bancaire qui s'élèvent à 1 500 €. Et ensuite, on a quelques goodies qu'on essaie quand même de limiter au niveau de la SFBI, ou en tout cas apporter un aspect écologique aux goodies. Donc en termes de recettes, au total, on a eu pour 11 200 €. Oui, 11 203 €. Et par contre, on a eu beaucoup plus de dépenses. Donc dans les dépenses, on a aussi des frais de gestion bancaire. On a les subventions de JOBIM 2025. On a les subventions de JOBIM 2026. Donc là, c'est d'habitude, les subventions des JOBIM se donnent après les JOBIM. Et là, en fait, cette année, pour JOBIM 2026, la subvention a été donnée 1 an à l'avance. On a aussi des frais d'assurance, puisque toute la conférence, l'ASFBI a eu également une assurance de 128 € par an. On a aussi des dépenses dues aux goodies. Ensuite, on a eu des adhésions SFBI, COSAF. On a les bourses de voyage de 14 000 €, incluant les prix. On a également les frais de communication et les missions SFBI. Et donc, ça nous a donné un total de dépenses de 40 072 €. Et donc, au 31 décembre 2025, donc, le compte de la SFBI était à 74 000 €, donc, ce qui représente moins 28 000 €. Mais nous n'avons pas encore reçu la clôture du JOBIM 2025. Donc, je vous rassure, tout va bien. Voilà. C'est déjà les résultats ? OK. Alors, maintenant qu'on a fait le point sur le bilan moral et le bilan financier, on va passer aux résultats du vote du bilan moral et du bilan financier. Donc, merci à tous ceux qui ont pris la peine de voter. Donc, en 2026, nous étions, donc juste avant l'ouverture des votes, 404 adhérents. Nous avons eu 125 votes, ce qui représente 31% de participation. Je pense qu'on peut faire mieux, avec seulement 2 abstentions. Et du coup, les bilans ont été votés donc positivement. On a eu 100% de yes et 100% de yes pour les deux. Donc merci à vous et nous avons voté. Alors maintenant, on va passer au renouvellement du bureau. Donc ici, je vous représente encore les membres du bureau actuel. Et comme je vous l'ai dit, donc, je suis actuellement présidente de la SFBI, mais ça fait 2 mandats, donc ça fait 6 ans que je suis à la SFBI, et il est temps de passer la main. Voilà. Donc je voulais juste revenir rapidement sur cette aventure, pour le partager aussi avec vous, parce que c'est une aventure qui a commencé d'une manière un petit peu cocasse, puisque j'étais présidente du comité d'organisation des JOBIM 2020, qui devait se tenir à Montpellier. Et tout le monde sait ce qui s'est passé en 2020. Donc on a dû faire des claquettes, comme on dit, ou en tout cas rebondir rapidement et trouver des solutions. Donc je ne sais pas si Vincent Lefort est là. Voilà, il est là. Donc merci à mon co-organisateur, co-président, parce que ça a été une belle aventure. Et on a réussi à faire le premier JOBIM en virtuel. Ça n'a pas été facile, mais on l'a fait. Donc, on va dire, le premier pied dans la SFBI a été un peu violent quand même. À l'issue de ça, j'ai rejoint la SFBI. Et puis, comme beaucoup de personnes, alors on me le dit de temps en temps, mais je ne comprends pas la différence entre la SFBI, le GDR-BIM et l'IFB. J'étais dans le même état que vous. Et du coup, j'en avais parlé avec Morgane et avec Hélène. J'aurais dit, les filles, il faut que vous m'expliquiez parce que là, je ne comprends pas. Et donc, du coup, je me suis intéressée à la SFBI et je me suis retrouvée à la SFBI. Donc faites attention à vous, ne posez pas trop cette question. On a beaucoup œuvré avec le bureau pour essayer de faire en sorte de clarifier les choses, parce que c'est quand même assez complexe, on se l'avoue, mais de faire en sorte de clarifier les choses pour la communauté. Donc on espère vraiment que pour vous, c'est vraiment beaucoup plus clair. Et vous voyez ce joli dessin ici au milieu qui nous permet un peu de résumer ça. Si vous avez d'autres idées, si vous avez d'autres questions, on peut aussi essayer d'améliorer les choses, ou voilà, on peut faire peut-être d'autres documents de soutien ou des vidéos. Voilà, faites-nous savoir. Et si vous voulez être bénévole, c'est super. Alors après, il y a eu un autre point, je vais dire chaud, de la SFBI, c'est le site web qui a brûlé. Alors, c'était le serveur OVH, plus précisément. Et donc, on s'est retrouvé sans site web. Donc, ça a été aussi une belle aventure, puisqu'il a fallu repenser le site web. Retrouver les vestiges du site web pour le refaire. Et là, je tiens à remercier énormément Julien Fumet et l'IFB parce que ça a été un gros travail. Aussi Morgane qui a recherché dans les archives les anciennes pages, les archives du site. Donc ça a été un gros travail. Et puis après, on a eu, c'est pas fini, hein. Après, on a eu, le Covid a continué. Donc en 2021, on a eu le JOBIM à Pasteur qui s'est refait en virtuel aussi. Et puis après 2022, on a enfin pu refaire un JOBIM en présentiel à Rennes et c'était génial. On était vraiment contents de pouvoir se retrouver, même s'il fallait bien faire attention aux distances sanitaires. Et puis en 2023, ça a été assez compliqué aussi, parce que du coup, on n'avait pas vu venir les choses. On avait l'ISCB qui avait prévu d'organiser une conférence internationale de bioinformatique à Lyon. Et là, il a fallu aussi être très réactif et trouver un moyen de continuer à organiser les JOBIM tout en faisant part à cette manifestation qui avait lieu chez nous, en France. Et donc, ça a été aussi une aventure très intéressante, puisqu'il a fallu du coup échanger avec l'ISCB. C'est comme ça qu'on est d'ailleurs devenu maintenant affilié. Mais on a eu aussi beaucoup de sueur froide avec Guy, Marie-France et Sandra pour organiser une session de bioinformatique in France dans la conférence ISCB. Donc ça a été aussi un gros travail, beaucoup de discussions, mais ça a été une belle réussite. Et puis après, comme Sandra vous l'a déjà dit, il y a eu le listing, on s'est dit comment aider les jeunes bioinformaticiens à ne plus être isolés, puisque ça a été une question qu'on avait eue, et puis l'ouverture à l'international. Donc tout ça, c'est voilà, tout ce que vous pouvez faire à la SFBI, en rejoignant le bureau de la SFBI, vous pouvez contribuer à vraiment animer notre communauté par différentes actions. Et du coup, pour ça, je tiens à remercier, parce que je ne l'ai pas fait toute seule, je tiens à remercier tout le bureau. Donc ici, j'ai fait le screenshot de notre dernière réunion de bureau. Donc merci. Merci à Delphine, Audrey, Charles, Sandra, Marie-France, Cyril, Pierre, Élodie. Merci aussi à ceux qui étaient là avant, et Juan. Merci aux anciens présidents, parce qu'ils m'ont fait confiance et parce qu'ils avaient déjà tracé la route, ils avaient fait un gros chemin. Donc merci à eux, parce que la passation s'est super bien passée, donc ça a été vraiment un vrai plaisir. Et je tiens à préciser que le bureau, ils ont été vraiment top, parce qu'il y a des jours où j'arrivais avec des idées un peu farfelues. Et ils m'ont quand même suivi, et donc ça, c'était vraiment très agréable. Donc merci à eux, et merci aussi à vous, toute la communauté, parce que ça a été un vrai plaisir de pouvoir rencontrer et discuter avec tout le monde. Alors c'est pas fini, hein, mais en tout cas, ça a été un vrai plaisir de pouvoir parler avec certains collègues avec qui on n'aurait pas eu forcément l'occasion en temps normal. Et donc merci pour votre confiance. Voilà.
Alors nous, on voulait en profiter pour faire un grand merci à Anna-Sophie, parce que ces dernières années, elle a quand même été présidente pendant 5 ans sur ses 6 années de mandat et vice-présidente pendant 1 an. C'est un travail énorme. Elle s'est vraiment dévouée à la communauté. Vous l'avez vu, elle a mis en place plein de projets dans un dynamisme exceptionnel. Donc vraiment un grand merci.
Alors, mais Sandra, t'en vas pas? Ouais, séquence émotion. Donc maintenant, je ne suis plus la présidente de la SFBI, et donc je vais passer la main à ma vice-présidente pour la suite et pour vous présenter le nouveau bureau de la SFBI.
Merci. Donc comme Anna vous l'a dit, cette année, on a 4 personnes sortantes. Donc Audrey Bioué, qui a donc fait un mandat au sein de la SFBI, Anna-Sophie, donc qui finit ses 2 mandats. On a également Pierre Poulain, donc, qui termine son 1er mandat, et puis Marie-France Sago, qui termine également son 1er mandat. On a 4 candidats. Donc Marie-France a proposé de faire un 2e mandat parmi nous. Et puis on a 3 petits nouveaux qui se sont proposés. Donc Jérémy Juste, qui est basé donc à Lyon, Emmanuel Morin, qui est basé à Nancy, et Emmanuel Tala, donc, qui est basé à Marseille. Donc j'espère que vous avez toutes et tous voté et que vous faites partie donc, des 125 votants. Comme le disait Anna-Sophie, on peut clairement faire mieux. Enfin, 30%, c'est vraiment, vraiment pas beaucoup. Donc on compte sur vous pour l'année prochaine pour motiver davantage tous vos collègues et la communauté. On aimerait au moins atteindre 40-45%. Je pense que c'est largement faisable. Donc voilà, et félicitations aux 4 petits nouveaux qui nous rejoignent. Je pense que vous êtes tous les 4 présents, donc si vous voulez venir, n'hésitez pas. Donc on est sur 404 membres qui pouvaient voter, 125 qui ont voté, donc avec un taux de participation d'à peu près 31%, et on a 2 abstentions. Donc félicitations à tous les 4 et surtout bienvenue à la SFBI. Donc voici notre nouveau bureau pour l'année 2026-2027. Donc un grand merci à la communauté pour soutenir la SFBI. Et on vous propose une présentation assez rapide, donc des points qu'on voudrait mettre en avant et qui vont nous occuper dans les quelques mois à venir. Et je passe la main à Charles.
Ok, bonjour à tous. Donc, c'est pas fini, c'est pas parce qu'Anna s'en va qu'on va plus rien faire. Donc, on a déjà quelques projets que je vais vous présenter en cours. Alors, d'abord, ça concerne les relations internationales. Donc, on l'a vu, on s'est rapproché de la communauté brésilienne et ouest-africaine, italienne également. Et puis, récemment, j'ai un truc pour pointer. En fait, récemment, la communauté canadienne s'est fédérée, donc littéralement, c'est-à-dire qu'ils ont créé une communauté fédérale. Donc, c'est un gros financement du CIHI. Et en fait, ils ont un représentant par province dorénavant, et ça s'appelle le Canadian Bioinformatics Hub. Et ils ont fait leur première conférence à Toronto en mai. Donc, la SFBI a été représentée. Et d'ailleurs, je tiens à signaler qu'ils ont pris JOBIM en modèle, c'est-à-dire qu'à l'ouverture, ils ont dit ce qu'on voulait, c'était faire un JOBIM, et donc c'était leur premier JOBIM à Toronto. L'année prochaine, c'est à Winnipeg, je crois. Et puis, un autre partenaire qui s'est manifesté à cette occasion, c'est aussi Génome Canada. Donc, c'est l'équivalent de l'IFB, en gros, pour eux. Donc, on se retrouve avec nos alter ego canadiens. D'un côté, la SFBI avec le Canadian Bioinformatics Hub, et de l'autre, on a l'IFB et Génome Canada. Donc, on va travailler tous ensemble pour essayer de développer des relations, donc des échanges scientifiques, des bourses de voyage. Enfin, voilà, on a plein d'idées. Tout ça, c'est en construction. On vous tiendra au courant. Et puis, la deuxième initiative dont on voudrait vous parler, c'est donc dans le cadre de l'année 2027 ADN dans tous ses états. Donc, c'est une année thématique qui est lancée par le CNRS, qui est co-organisé par le GDR-BIM et le GDR-IFM, Informatique fondamentale et ses mathématiques. Et puis, dans ce cadre-là, la SFBI a proposé de monter une collaboration avec Métal Hurlant. Alors, pour les plus jeunes, Métal Hurlant, c'est un mythe. Donc, c'est une revue qui date des années 70, qui a vu les plus grands noms de la BD française, Druillet, Moebius, puis plus tard Bilal, Mézières, enfin bref, ils y sont tous passés. Et en fait, on les a contactés et on leur a dit « Oh, au fait, le CNRS lance un truc sur l'ADN, ça vous dirait de faire un numéro spécial ?» Et dans les 3 jours, ils ont répondu. Donc, ils sont hyper motivés. On a reçu... Donc, l'idée, c'est de créer des équipes qui associent des chercheurs avec des scénaristes et des dessinateurs pour créer des histoires. Donc, un numéro de Métal Hurlant, je vous invite à acheter le dernier numéro. C'est à peu près 25-30 histoires. Donc, ils vont sélectionner... On a eu 50 propositions de chercheurs, plus de 50, 51. Et là-dedans, ils vont sélectionner quelques histoires pour s'inspirer de ça. Donc, la partie création, ça sera côté Métal Hurlant, mais l'inspiration viendra de la bioinformatique. Donc, ce n'est pas de la vulgarisation scientifique. Vous n'attendez pas qu'on vous explique ce que c'est qu'un sujet, etc. Ça sera au-delà de ça, c'est un peu plus artistique, c'est un peu plus créatif, on espère, on verra. Donc le numéro est prévu pour mai-juin 2027, donc normalement l'année prochaine, à cette période, le numéro sera sorti. Voilà, donc c'est les 2 initiatives en cours. On en fera d'autres parce qu'on veut démontrer à Anna-Sophie qu'on peut s'en sortir super bien sans elle, en fait. Voilà. Et je passe la main à Sandra, de nouveau.
Oui,
Non.
juste 30 secondes. Donc, comme on vous l'a dit, en tout cas, on a essayé de vous faire passer le message, la SFBI est une vitrine, mais elle ne travaille pas seule. Et là, on voulait vous montrer notamment une collaboration entre le réseau Mérite et la SFBI et sur la mise en place en fait, d'une enquête sur les conditions de travail des bioinformaticiens. Et du coup, je vais laisser Erwan présenter cette partie.
Merci Sandra. Donc, bonjour à tous. Donc l'idée, c'est juste avant de vous lâcher devant la pause café, donc de vous faire un retour très rapide sur cette enquête, donc qui, comme le disait Sandra, a été ouverte au mois de février et qui vous a laissé 1 mois et demi à peu près pour pouvoir répondre à un certain nombre de questions qu'on avait construit au sein du réseau Métier Mérite et au sein du bureau de la SFBI, avec l'aide, il faut le noter, j'ai manqué de le préciser ici, mais avec l'aide finalement d'un travail qui avait été préalablement fait au sein de la communauté Info Biodiag, qui avait mené également il y a 2 ans de ça une enquête et dont on s'est quand même grandement inspiré pour pouvoir interroger une communauté cette fois-ci un peu plus vaste. Donc un grand merci aux 250 répondants à cette enquête. Et l'idée, c'est pas de vous faire un retour exhaustif là, maintenant, pendant ces quelques minutes là qui nous sont accordées, c'est de vous indiquer un peu un retour sur la cartographie des répondants et de vous faire un tout petit bilan très rapide sur ce qui est ressorti de cette enquête-là. Donc, en termes de cartographie des répondants, on est plutôt satisfait sur ces 250 répondants. On a conscience qu'on aurait pu arriver à mobiliser encore plus. Il y a 500 personnes à JOBIM. Là, c'est la moitié de la salle qui a répondu. Mais on est quand même malgré tout assez satisfait parce qu'en termes de cartographie, on a une répartition assez équilibrée du sexe ratio. On a une couverture assez homogène des tranches d'âge. On a, en termes de niveau d'études, on a un niveau d'études qui correspond finalement à la population des bioinformaticiens, qu'il s'agisse d'ingénieurs, de chercheurs ou d'enseignants-chercheurs. On s'adresse à un public qui est majoritairement Bac+5 ou Bac+8, donc c'est assez cohérent. Et puis, en termes de répartition finalement du domaine scientifique de formation initiale, on retrouve majoritairement des gens qui sont des répondants qui sont issus de formation initiale en bioinformatique, ce qui dénote finalement des répondants qui sont relativement jeunes, puisque si on s'adresse à une communauté plus âgée, on va retrouver plutôt des gens qui ont une formation initiale en biologie ou éventuellement en informatique, qui ont versé du côté obscur de la force plus tard. Mais malgré tout, donc, on retrouve une communauté de bioinformaticiens et de biologistes, et malgré tout des informaticiens, des mathématiciens, et un peu moins d'une communauté de chimistes. En termes de statut, encore une fois, une forte représentation des ingénieurs et peut-être un sous-échantillonnage des chercheurs et chercheuses et maîtres de conférences. Alors là, on doit battre notre coupe. En fait, on a été plusieurs à lire les questions qui vous ont été adressées et on s'est rendu compte qu'on avait eu un trou, un moment dans la matrice, et notamment, on a peut-être oublié de poser des questions qui étaient peut-être un peu plus spécifiques à la communauté des chercheurs ou des enseignants-chercheurs, notamment par exemple sur les commissions de recrutement. On s'est beaucoup focalisé sur les BAP qui concernent beaucoup plus les ingénieurs, mais peut-être qu'il y avait des questions autour des commissions de recrutement, et là il y a peut-être des choses, des discussions à poursuivre, peut-être avec, je te regarde, mais au niveau du GDR ou d'autres membres de la communauté pour pouvoir percevoir peut-être des dissensions, des décalages qu'il pourrait y avoir. En termes de type de contrat, on a une répartition, encore une fois, qui est équilibrée entre les titulaires, les CDD, les CDI. Et en termes de régions dont étaient issus les répondants, une grosse représentation de la région Île-de-France, qui est quand même assez attendue, une bonne réponse au niveau de la Bretagne, des Pays de la Loire et de la région Auvergne-Rhône-Alpes, et peut-être, et on le regrette un peu ici, parce qu'on sait c'est une communauté qui est très active, un sous-échantillonnage de la communauté Provence-Alpes-Côte d'Azur, parce que c'est un petit peu triste finalement d'avoir que 17 personnes qui viennent de là-bas, alors qu'on sait que c'est une communauté qui est très active. En termes de retour, alors le message globalement positif de cette enquête, c'est que globalement, répondants, répondantes sont quand même satisfaits ou très satisfaits finalement de leur situation. On a une communauté qui le matin se lève, et va au boulot et sait pourquoi il va au boulot et sait qui travaille pour quelque chose et il fait quelque chose qui lui plaît. Donc ça, c'est quand même un message qui est quand même globalement positif, très positif. Mais à côté de ça, on a relevé malgré tout un certain nombre de préoccupations qui ressortent autour des problématiques de précarité croissante de l'emploi contractuel, des difficultés qui est couplée également à des difficultés accrues en ce qui concerne l'accès à l'emploi permanent. Et malheureusement, ce n'est pas les messages que nous ont transmis nos instituts et ESR respectifs qui vont qui vont rassurer actuellement les contractuels, mais bon, on va quand même travailler à développer quand même cet aspect-là avec les pôles RH et avec nos instituts. Une reconnaissance technique qui est réellement avérée, mais quelque chose qui est couplé finalement à la première préoccupation, une reconnaissance institutionnelle, quand il s'agit de contractualiser les postes, qui est beaucoup plus difficile à obtenir. Un écart de rémunération persistant entre les secteurs publics et privés, qui n'est pas spécifique à la communauté bioinfo, mais qui est particulièrement vrai quand on s'adresse à la communauté plus informatique, on va dire, où là les décalages peuvent être réellement considérables. Et un isolement technique qui peut être parfois ressenti pour des bioinformaticiens qui sont un peu isolés dans les équipes et qui ressentent un besoin renforcé en formation continue. Et dernier point, une classification des métiers d'ingénieur dans des BAP qui ne reflètent pas ou pas parfaitement finalement le travail à l'interface entre deux mondes, cette interdisciplinarité qui est réellement propre au travail des bioinformaticiens et sur lequel il nous faut travailler avec nos instituts. Donc, ce n'est qu'un snapshot très rapide de cette enquête. Vous pouvez avoir une vue un tout petit peu plus exhaustive au niveau du poster, donc qui est positionné sur le poster numéro 2 et qui est présent sur toute la durée de l'événement. Et on a tâché sur le site web de Mérite, dans la rubrique GT Métiers, de faire une transcription un peu plus exhaustive de l'enquête, avec une synthèse sur des points qui nous semblaient des points un peu importants, qui avaient transparu au moment de l'analyse de l'enquête, un certain nombre de graphes aussi qui synthétisent l'ensemble des questions. Alors, il y a quelques bugs, quelques bugs d'affichage, on le sait, on va travailler là-dessus, mais vous pouvez retrouver l'ensemble de l'information ici. Donc voici pour l'enquête. Comme les organisateurs ont eu l'erreur de me confier le micro, je vais continuer à le garder pour vous présenter Mérite. Et je vais tâcher de rester un petit peu dans les temps. Donc, Mérite, comme vous le savez, et comme ça a déjà été présenté par Sandra il y a quelques minutes, est une des structures d'animation, alors c'est une autre manière de présenter ces structures d'animation de la communauté, mais une des structures d'animation de la communauté qui se positionne réellement sur les activités d'ingénierie en bioinformatique, mais pas uniquement pour un public d'ingénieurs, ça peut être également pour un public de chercheurs intéressés par des questions d'ingénierie. C'est un réseau national d'ingénieurs en bioinformatique des établissements supérieurs de la recherche, donc des ESR. Ce n'est donc pas un réseau qui est limité, même si ça a été initié au niveau du CNRS, ce n'est pas limité à cet ESR. Ça se veut être définitivement un lieu d'échange entre bioinformaticiens pour réaliser de la veille scientifique et technique, pour faire du partage d'expertise, pour faire des retours d'expérience. Et comment on fait ça? On fait ça en organisant des groupes thématiques de travail sur des sujets sur lesquels je reviendrai tout à l'heure, qui organisent des webinaires, qui organisent différentes actions tout au long de l'année, des événements nationaux qui peuvent être des journées scientifiques ou technologiques, des formations, des ANF, ou encore du soutien, et c'est une des activités clés des réseaux métiers du CNRS, du soutien aux réseaux locaux ou régionaux au travers de conseils, de retours d'expérience et également d'un soutien financier. Le comité de pilotage est constitué de 8 membres, donc Vincent, qui est coordinateur du réseau métier, et 7 autres membres qui sont localisés dans différentes ESR, donc Audrey, Hélène, et moi-même, Vincent Lombard, Alexandra, Loredana et Jimmy. On est 7 ici présents, donc n'hésitez pas à venir vers nous pour revenir discuter avec nous de ce réseau si vous avez des questions concernant l'organisation de celui-ci. Et on définit nos membres en tant que, comme les personnes qui sont inscrites à la liste de diffusion au Mérite. Donc si vous ne l'êtes pas encore, et certains d'entre vous ne doivent pas l'être, puisqu'on est actuellement à 250 abonnés, on était à 200 l'année dernière, me semble-t-il, me disait Vincent, et qui sont répartis dans différentes tutelles, CNRS, INRAE, CEA, CIRAD, etc., avec des membres qui sont majoritairement dans la communauté des ingénieurs de recherche, ingénieurs d'études, mais également des membres qui peuvent être chercheurs ou enseignants-chercheurs. Parmi les activités, donc, une des activités, c'est de soutenir des réseaux locaux ou régionaux. À l'heure actuelle, on a identifié à peu près 16 je crois, réseaux locaux, dont certains sont des réseaux qui sont très clairement établis et que vous allez pouvoir retrouver sur une carte dynamique sur le site web de Mérite avec les liens qui correspondent et les contacts qui correspondent à ces réseaux. et des réseaux qui sont actuellement en cours de structuration avec lesquels nous discutons pour pouvoir les aider justement à mettre en place des actions structurantes. Et parmi les réseaux qui sont actuellement en phase de structuration, on peut citer notamment en Normandie où on avait un trou sur la raquette jusqu'à présent, un des trous sur la raquette du territoire français, qui est actuellement en train de se structurer à Rouen et peut-être par la suite à Caen. Et on incite très fortement nos collègues de Bordeaux ou de Toulouse également à mener des actions de coordination ou d'animation au niveau de ces régions. Et si vous avez entamé une action de coordination et que vous n'êtes pas représenté sur la carte, n'hésitez pas à revenir vers nous. Les groupes thématiques dont je vous parlais tout à l'heure, il y a 2 ans de ça, quand on a monté le réseau métier, on a interrogé la communauté sur les actions, en tout cas des groupes thématiques qui semblaient être intéressants à mettre en place au regard des besoins de la communauté. Il y a 7 groupes thématiques qui ont été identifiés et qui ont commencé à mener des actions au cours de ces 2 années. Je ne vais pas rentrer dans le détail de l'ensemble des actions, mais de l'ensemble des coordinateurs que vous allez pouvoir retrouver ici sur cette slide, mais également sur le site web du réseau Métier Mérite. Mais on retrouve donc un groupe autour des questions du fer, de la reproductibilité et des workflows, du single cell et de la transcriptomique spatiale, des long grids, de la notation, de l'intelligence artificielle et de l'intégration de données hétérogènes, auquel s'est rajouté également un groupe autour de la reconnaissance du métier de bioinformaticien. L'ensemble de ces groupes ont été très actifs, vous avez dû le recevoir dans les listes de diffusion de la SFBI, et ont mené soit des actions, des webinaires qui ont été organisés de manière très très régulière et très, très fréquente pour certains des groupes de travail, et ou des actions de formation ou encore des mini-symposiums, et c'est le cas notamment demain avec le groupe Annotation qui organise en collaboration avec le PEPI EBIS Annotation, le PEPI Annote, pardon, des PEPI EBIS, un mini-symposium sur la notation des génomes eucaryotes, la notation structurale des génomes eucaryotes. Dernière chose, dernière slide, un petit bilan sur les actions passées et celles à venir. Un des événements marquants sur l'année qui vient de s'écouler, ça a été l'intégration officielle du réseau Métier Mérite au sein de la MITI, donc de la Mission pour les initiatives transversales et interdisciplinaires du CNRS. Jusqu'à présent, le réseau Métier Mérite avait été soutenu par différents instituts du CNRS. Et là, depuis le mois de décembre, oui, c'est ça, décembre 1er janvier, pardon. Donc, depuis le mois de janvier, donc c'est acté pour 4 ans. Le réseau Métier Mérite fait officiellement partie de la MITI. Donc à ce titre, on a des grands pouvoirs, c'est-à-dire des financements, mais également des grandes responsabilités, parce que ces demandes de financement, elles ne peuvent pas être faites au fil de l'eau. Il nous faut pouvoir réaliser une demande annuelle qui va être votée et validée par la MITI. Et donc, pour vous, si vous souhaitez que le réseau Métier Mérite vous aide dans des actions locales, dans des actions des GT autour des questions qu'on a évoquées tout à l'heure, il faut revenir vers nous très vite afin qu'on puisse budgéter les sommes nécessaires et les faire remonter au budget que l'on va demander à la MITI. Donc tout ça nous aidera à faire des montages pour des actions nationales et ça nous offre également la possibilité de pouvoir interagir avec d'autres réseaux métiers, notamment le réseau Devlog ou d'autres réseaux métiers qui nous ont d'ores et déjà contactés. On a mené l'année dernière, donc, à l'aide notamment de l'IFB et d'INRAE, donc une ANF qui était sur les workflows et la reproductibilité, qui a réuni pendant 3 jours 20 participants encadrés par 15 personnes, une équipe pédagogique, donc à Villejuif. Les supports sont accessibles et vous pouvez les consulter. Et l'enquête de satisfaction semblait plutôt bonne au regard des retours qu'il y avait eu. Donc de ce fait-là, on a décidé de redéposer un projet d'ANF qui a été validé par la MITI. Enfin, en tout cas, cette proposition d'ANF a été validée par la MITI. Maintenant, ça doit suivre le cycle des ANF. Et le sujet de l'ANF va être développement d'analyse bioinformatique reproductible à l'aide de l'IA générative. Et enfin, sur les actions, en dehors des actions sur les GT et des groupes de travail locaux, une des actions majeures qui a été menée par le réseau métier, ça a été la mise en place de cette enquête. Et au-delà de la mise en place de cette enquête, ça va être d'utiliser les résultats de cette enquête pour remonter finalement ces besoins de la communauté auprès de la MITI et auprès de la DGRH du CNRS. Et ça, c'est un travail qui est actuellement en cours, puisqu'il y a une réflexion qui est menée au niveau des CNRS sur le métier de bioinformaticien. Et je vous remercie de votre attention.
Merci beaucoup, Erwan. Malheureusement, on n'a pas vraiment de temps pour les questions, parce qu'on a été un petit peu long et un peu bavard. Donc là, c'est l'heure de la pause. Par contre, les membres du bureau de la SFBI, en grande partie, sont disponibles sur le stand pendant tout le long de la conférence. Donc si vous avez des questions, des remarques, n'hésitez pas à venir nous voir. Et puis, comme l'a dit Erwan, vous pouvez retrouver aussi les membres du réseau Mérite tout au long de la conférence. Merci.
plusieurs micros, vous allez vous
Ouais,
passer,
parce
hein.
qu'on fait un chœur.
Ok.
Hello, tu me dis quand tu veux qu'on commence?
On a combien de temps encore?
20 minutes, mais...
Question comprise, donc on a misé sur 15, c'est ça?
Ouais. 15, mais on a 25 minutes.
Mais les 5 minutes, c'est 5 minutes avant 15, c'est ça?
C'est au bout de 10, quoi.
Carrément, ouais.
C'est complexe avec nous, hein. Bah
Allez.
si, bien sûr, vas-y. Je te laisse pas... Tu fais peur. Bah oui, c'est le jeune Jacques. C'est bon, il y a du monde?
Bah ouais, il y avait personne.
J'ai fait de la pub, hein. Mais là, il y a de l'air. Ah bah oui, il y a le ventilateur. Ah bah voilà, c'est vrai qu'il faut ça. Okay. Thank you, everyone. One question. We were wondering if we should switch to French. Is there anyone that absolutely needs this in English?
Who prefers?
No, yeah, two, yeah. Okay, so we'll continue in English. So thank you everyone for coming to the presentation of the Institut Français de Bioinformatique. So we are called IFB Elixir France. So I'm here with Jacques Van Elden and Anne-Françoise Adam-Blondon. My name is Morgane Thomas-Chaulier and we are part of the direction team of Institut Français de Bioinformatique. So very quickly, the IFB is a federation of bioinformatics core facilities and teams. So we are decentralized all over France, and we are made of 20 member core facilities and 16 associated core facilities, but also research teams, and one coordinating unit. Just to be clear, IFB and the French Elixir node, so Elixir FR, is the same thing. And indeed, we have an inclusion into Europe, and we are part of Elixir. And there are more than 200 ETPs, so more than 400 experts that are working at IFB. So many here at JOBIM are actually working with us. So very quickly, we are a key player in French and European bioinformatics. We are what is called a research infrastructure. In French, it's infrastructure de recherche. And we provide services and we conduct R&D projects. And actually the talk of Christophe Decimos was really good to introduce all the topics that we are working in at IFB with sovereignty of data and digital as well, training. We are in open science and AI. So various key figures, just to give you a short idea in numbers for last year. We have more than 13,000 active users of our HPC services and computing resources. In our core facilities, more than 100 projects have been accompanied, and we have trained more than 5,000 people just for last year. So we are going to follow some users, and we are going to present you Marc. Marc is a neurobiology researcher, and Marc didn't do his project really well because he started by generating his data, and he has no expertise in data analysis or bioinformatics, and now is in search for help. So next time we would advise Marc to actually pair directly with a platform, but luckily we have a national network of core facilities that offer as services analysis service, and some teams and core facilities are specialized in specific teams depending on their local environment. For example, in Roscoff, it's no news that they work on marine biology. Let's follow Sonia. Sonia, she's a marine biology research director. Maybe she does work in Roscoff. And she's preparing a very ambitious project, something like a PEPR. And she knows she's going to generate lots of data, and she needs to strengthen her project, and so she's really looking for a partner for the project that will be able to deal with the problems of computing, storage, analysis, and so on. So she needs to find a collaborator within our research infrastructure. And we are partner of 8 PPR Grand Défi, so we are able to partner on very large projects, including European projects. Hugo is a second-year PhD student. He's working in plant biology, and he has generated genomic data in agronomically important plants. So he has a bioinformatician in his unit, so he was not really worried, but the person is overwhelmed. Probably you know this case. And he's still early career. His professor told him, well, you should study bioinformatics, and so he needs training. And this is something that we are really strong in at EFB, in training. Just for in-person training last year, we had more than 2,000 people train with us. And we have on our website a catalog of courses, and each time, as we are the French node of the Elixir research infrastructure, we also provide European-level resources. And our core facilities, and at the national level, we propose some courses. The issue is that Hugo has to be quick in gaining skills, so he's going to do some self-training while he's waiting to actually go to his course. And luckily, at EFB, we propose some e-learning. In particular, we give access to all the pedagogical material from our past session. And recently, we have made up some online tutorials thanks to the group e-formation. And at the European level, there are also lots of resources. On the Moodle part, there is also training for bioinformaticians. So if you want, have a look. There are advanced topics, in particular in AI, in open data science, single-cell analysis. And for the e-formation, we have a fresh publication, so it's a bit of advertisement, on the work that we have been doing recently. So Penelope, she's a professor in microbiology, and she knows how to analyze her data. She was trained by EFB, so she knows what she's doing. But she struggles to install software tools on her laptop. And in addition, she wants to work with her students, and she wants to propose a practical with her students. And she needs an access to server where the tools would be already installed with account for her students. And at EFB, we do provide this infrastructure. So we have 15 sites that propose high-performance computing for life science. So they are either local, and we also have 2 mutualized core sites that are national, and that you see on the image. They are in orange. And we propose 2 complementary technologies, the cloud with virtual research environments, and the cluster. And you can access the cluster with the SSH approach, but we also have a more, let's say, non-expert friendly way, which is a web-based browser with open on demand. So we also provide user support. We have project spaces for multi-users and also for students. And this is managed by a distributed team of experts from the core facilities. And you have the numbers on the right to show you that we do have lots of equipment. In addition to the cluster and the cloud, we propose 1 national mutualized site of Galaxy France. So this is a very strong and important portal. It's a simplified access, so this is also for biologists, but not only, because you can also run workflows with Galaxy. And it allows to do some reproducible analysis. And this is a way also to showcase some tools that are developed by French teams and also in Europe. So the Galaxy persons that work with us at EFB are very active, and this year they just co-organized the community conference of Galaxy in Clermont-Ferrand. So that illustrates that we are really heavily involved in Galaxy. So what Marc, Sonia, Hugo, and Penelope have in common is that they all have generated research data. So they need actually guides, tools, and help to comply with the data openness policies. And the thing is that it shouldn't be too time consuming because it's a pain for the researchers to actually comply with all this. But we do have a strong emphasis on open science for data, and we provide training like Fair Data and Fair Bioinfo and guides on our websites, lots of guides on the European level as well. There are some core facilities that have specialty in helping on this part, like URGRI for plant data. And within EFB, we do develop tools. So some of you were yesterday at the demo of MatBot. And we also have, for example, Fair Checker for compliance with FAIR principles. Finally, Sophie, she's a bioinformatics software developer, and she developed bioinformatic tools, but she would like more biologists to use her tool, so she needs to make it more accessible to the users. And for that, we do propose also some resources. So it's possible to install the tool on our computing infrastructure. Also referencing, we provide a catalog of resources in France, and we also have the Elixir label, and it's possible to apply to this label, and we are in charge of that. And we also provide skill development for bioinformaticians. And last but not least, Romain. So I like Romain. Romain, he's a support engineer. Romain is going to soon leave his lab for a new position, but he was the one managing the lab server. Probably you know that there are some labs that have their own server. The server is not under warranty anymore. The person didn't have any time anyway to continue to install the tools. And before leaving, Romain, he would like to find a solution for the lab so that he lives, let's say, freely. And again, that's where we propose our computing infrastructure. But here I want to emphasize the fact that it's already ready to use. On the cloud, we have 65 virtual environments that are already ready to be deployed in one click. On the cluster, we have more than 800 tools, 500 Air packages that are already installed. On Galaxy, it's more than 1,700 tools that are already there. And this is open for everyone. So no need to continue to have the lab server. And I'd like to emphasize the fact that there are more than 20 experts. Some are in this room today. Engineers from core facilities that are sharing their expertise, their time, their development, their installation recipes, so that they don't reinvent the wheel and they work together. And this is really an illustration of how mutualization can work. And it's also working outside of EFB, so there are 2 sites that are also joining the federation in France and outside of France. So I'm leaving.
So since this year, we defined 5 strategic axes, and what we define as a strategic axis is something where we have huge challenges that are really exceeding the IFB scope, actually, that are including us, but not only, where we are already active, we have expertise, we have actions, we already have some activity, but it's not enough, and we need to go one level up, and also where there is some urgent need to develop things. So one of them, well, I will not list all of them, I will just give some examples, but one of them is the digital infrastructure. Morgan showed where we are. Comment ça marche ce truc là ici? Comme ça? Ouais. Okay. Data resources, I think you had all the presentation by Christophe, who showed really brightly that this is a real huge challenge for all the scientific community. I will not come back to it. Now, the processing of sensitive data. The first thing you think when we say sensitive data is health data, and this is quite important. We have huge challenges there, but it's not only for health. Actually, the sensitive data includes also environmental data, biodiversity data that can be, well, where is some pirating risk, as Christophe said, and also some data that has an economical value, agricultural genomes, and so on. And so these are sensitive data. But of course, the first one on which we are active is on health data, because now there is a new engagement of France, a commitment of France to be part of what is now defined as the European Health Data Space, EHDS, where all the countries have a commitment to share the health data, the data that is produced for care, but also to share it for research. And actually, nobody is ready for that. So the Ministries of Health and the Ministry of Research are defining work groups in order to see how will France do that. And we are actively participating in this. Also at the national level, we are collaborating with INSERM and data centers in order to deploy bioinformatics resources inside health certified data spaces. About AI, we started in different context activities, but these activities are about benchmarking, testing the AI capabilities, and also training, organizing workshops. And we have some publications, we organize some hackathon. Recently we organized in June a hackathon where about 30 people came, and each of them could propose some study case, and then we addressed 10 of these study cases, and we worked during 2 days in order to try solving or testing how good are the AIs to solve these study cases. We also organized a collaboration with Université Paris-Cité and Merit, a one-day symposium about how to use AI for software management. No. Yes? Okay, and I will go fast. So orchestrating data flows through their life cycle. We will present with Thomas, who is there tomorrow, a mini symposium on that. But basically, so I invite the people who are interested to come here to participate to that. And I will let Anne-Françoise present the rest.
Okay. So super fast. So we belong also to the Club des INBS, so it's a club gathering all the national research infrastructure for life sciences in France. As you see, half of them are health-oriented, and others are technology-oriented, and we are the only one in bioinformatics. We have big challenge ahead of us, and we are trying to have one voice towards the Ministry of Research to have a consistent offer for the research in France for the next roadmap, French roadmap of the infrastructure. We have been working with many of these infrastructure. These are some examples of co-development of services between two infrastructure. I won't go into details, and it's only example, not all the landscape. We are also working with the French Ministry ecosystem that operationalize its policy on open science. So it's Recherche DataGouv ecosystem, and we are one of the thematic center of skills to support data stewardship for research in life science. And we do this, we do this also with the other French RIs. And so we are, you see, well, maybe I come back. A new incomer at the European level is EOSC. Perhaps you have heard about this European open science cloud that should give to any researcher environment of analysis and data storage, etc. And with all these ecosystems, we are trying to find our path and see how we will work also in this new landscape at the European level. So the challenge is really about sustaining research infrastructure and having a discussion with the ministry about that. We have short-term problems on fundings, all the research infrastructure, but we are working together to find solutions, and we need to find our path in EOSC in this new landscape. And I think this is all. No, pardon, sorry. So yes, meet us at the booth.
Questions?
Hi, I was wondering, for teachers and students in training, are there any restrictions on who can ask for an account or a series of accounts for practical sessions, learning Unix and bioinformatics tools and Galaxy and all these sort of things?
So as of now, we are open to academic users. So as long as they work in a university, there is no problem. We have actually been funded for 10 years, and that's why we could offer it up to now for free. And it's possible both on the cloud and on the cluster. We cannot assure you that this is going to be able to continue. Other questions?
Hi, sorry, I'm asking the same question every year. Now that you have set up a wonderful infrastructure for teaching, is it possible to have something that will be a diploma for people who are just willing to get a certification on one of the aspects of the huge bioinformatics library? So there are very nice courses on IFB, but the demand I see from many persons already working is getting that.
So there are some ideas on that part, but I'm going to be purely honest, and under the supervision of Hélène, we need help in terms of HR. So please come help us and make it a reality. Hélène says yes. Okay. Jacques really wants to say something.
Yeah. Actually, I skipped the future for AI, so since we have 37 seconds left, I would like to mention that actually AI is a big, big challenge that encompasses everything, because it encompasses the AI readiness, which includes correct metadata annotation. It requires also a strong physical infrastructure, digital infrastructure, software environment, and so on and so on. So we address this as IFB, but also in collaboration with higher level partners. And in particular, there are data centers in France who are specialized for AI, for the infrastructure. And in particular, Idriss has the Jean Zay supercomputing. Now in Montpellier, there is Adastra. But also, there is a European AI factory federation, and France is entering into this AI factory. And we started just to discuss with these people, because actually the needs will be much, much over what one national infrastructure can deliver. But on the other hand, we are the most in the best position to bring life sciences into this huge supercomputing. And also we are very aware about the environmental consequences and impact. And so we also have a work group on sustainable development and parsimony and so on. And we try to do the right balance between efficiency and cost.
Thank you very much.
Hi, so my name is Guillaume Fertin. I'm a professor at Nantes University. I'm a computer science professor. And I will... OK. Super, merci. And I will tell you about what's been already explained a little bit by Anna-Sophie about the GDR-BIM. So I wrote my slides in French, and I will pretend I did it on purpose so that the non-French speaking people in the audience can learn written French as I speak English. Okay, so what is it exactly? GDR means Groupement de Recherche, and BIM basically means molecular biology with a strong focus on methodology. We are funded by CNRS. When I say we, it means that CNRS provides some amount of money to my research facility, and I volunteer to be head of this GDR but I'm not being paid for that on top of my salary. And actually, so what we do, what I would like you to remember from my talk is, say, 4 things. The first thing is that JOBIM is mainly people making it work. And what do they do? I will explain that later. What do? They mainly what they do is that they organize scientific workshops, and they organize scientific school like summer schools. And what we do also, so this is the first thing. The first thing is JOBIM is people. The second thing is we organize things. The third thing is we provide money for you to organize things also, and for you to travel for the young people. I will be more specific a bit later. And the last thing is I would like you to subscribe to the web page, which is, I hope, no, I forgot. So I will let you know about it later. I'm lost in my slides now. Okay, I'll get back. Okay, before I go any further, so we're talking about research, about methodology. We are being funded by CNRS Computer Science, but of course it's not limited to computer science. We know we are talking biology, but there are also mathematicians, there are also chemists, and wherever you come from, CNRS, university, INRAE, INSERM, Curie, Pasteur, CEA, etc., you're more than welcome, of course. And whatever your position is also, whether you're junior, senior, postdoc, PhD, research engineer, researcher, assistant professor, etc., etc. So it's open to everyone. I have a challenge for you. As of today, we are 645 people listed on the group. I would like, by the end of the afternoon, that we are 650, and I think it's feasible. Last year, my goal was 600, and now it's more than 600, so I would like us to be more than 650 this year, maybe 700 if you all apply today. Okay, so people, so there are, the way it's organized, maybe it's not so important, but it's more or less 25 people discussing science and organizing things. And the real people who make the real work are the ones who organize what we call the groupe de travail. So each groupe de travail has a specific thematic focus topic. And I will list them very, very briefly after that. And as I said, if you want to subscribe, you just type my GDR on your favorite research engine, and you will find this page, and you just go there, you subscribe, it takes 5 minutes, and you decide which groupe de travail you want to belong to, and that's done. And we have reached the 650 people. So the groupe de travail, there are 6. I will not read everything which I put here. It's just that they meet every year. So when I put something in blue, it's for the future. If there's nothing in blue, it means that they've already met recently. So Alfie will meet in January next year in Paris. BIOS just met 1 month ago, but they have webinars. So if you're interested in systems biology, then you can apply there. Alfie, I forgot to say, is about phylogeny, evolution. We have a third group, which is LEGO. It's machine learning for genomics, and they will meet in December in Paris. The call is not out yet, but they plan to meet in Paris next December. Sorry. Massim is about structural biology. They met recently also, but they have a school. I will let you know about this later. SecBIM is about sequences and algorithms for sequences, and they will meet in Montpellier in November 26. And to finish with Statomic, so Statomic, Christelle is here, is one of the person responsible for the groupe de travail, and they already met at the beginning of the year, this year, but they will meet next year. What we do also is schools. So we have 2 schools that we organize. One is about statistics, algorithms for sequences, and the other school is about structural biology. So the first one on the top just took place 1 month ago, and next iteration will be in 2 years. And AlgoSB, they will meet in November, and I think you can still apply if you want. Okay, so that's what we do. That's what we really do. People organize these things. And we also, so this is what we do every year, more or less. We also do things which are exceptional things in some sense. Charles mentioned that. We did with another GDR, which is pure computer science. We decided to do something which is called ADN dans tous ses états. And it's about DNA, of course. There are 2 people who are running this. There are Camille Marché, maybe some people know Camille Marché from Lille, and Charles Paperman, who's also from Lille. And the idea is they asked a committee of experts to join them to think about what the research is about DNA in all what that represents, DNA and computer science. And the idea is that at the end they have to write, it would be good if they wrote a report, but they have 2 years to do that, and they can also animate the community. And Charles already mentioned this Métal Hurlant project. So Camille and Charles are member of that project. And I wanted to mention something which will appear soon. They organize also a day, jeunes chercheurs, jeunes chercheuses, about computer science and links with biology in October in Paris. So if you're interested, then you can have a look at the website, and you also can suggest a talk there. The deadline is on 7th of September. What we do also is with another GT who is called RT, Réseau Thématique, in ecology, evolution, RT Greenomics. We organize a 1-day gathering in Paris again, in October again, 2nd of October. Basically, it's AI for omics. So if you're interested, also, you can go there. I think if you're listed on the BioInfo mailing list, you got this information maybe yesterday, something like this, and the one about the previous one also has been sent recently. Sorry, so this I went back. Maybe what's most interesting for you is that GDR can give you money. We support some events. I listed a few here. The main support that we give in terms of amount of money is JOBIM. So GDR BIM is a big partner of JOBIM. And we support also any scientific gathering which is within the scope and which takes place in France. Usually we send also twice a year, also via the BioInfo mailing list, a call for support. If it's not easy for you to reach these 2 deadlines, December and April, you can still send us an email and we can discuss. We also support something which we would like to be more successful, is some money that we would give to a small group about an emerging subject. And in that case, if it's accepted, it will be supported for 2 years. The idea is you have identified a subject, a scientific subject, which is not covered yet or not fully covered, and you think it's interesting to push it further, then you can apply. And if accepted by the scientific committee, then you will be supported for 2 years. And you can ask anytime, au fil de l'eau. Same thing for visits. So this is a classic one. If you're a young researcher, so it starts at the PhD level and it stops where even if you're a permanent researcher or assistant professor after, say, 2 or 3 years, and if you need a small support to go visit another lab in France, then we can provide some support. again, you just send us an email, and if you also look at our website, we simplified the procedure, so it's not very hard to apply. So this is not things we directly do, but we help you do things. And to finish with something which is a bit exceptional, of course, 2026 is the 20th birthday of JOBIM. So we will organize in Paris, again in October, just after what's organized by Camille and Charles about ADN dans tous ses états. Just 2 days after, we organized the 20 years of the JOBIM. This will be a scientific gathering with some keynotes, which I listed here. So maybe you know those people, I won't list them exclusively. It's free, it's limited, so you can apply now. You can apply until the 15th of September, but if we are too successful, then we will have to stop at some point. So don't hesitate also to subscribe. So to finish with, this is my take-home messages. So this is open to everyone. You just go to this famous MyGDR webpage and you subscribe. As I said, we organize scientific workshops, summer schools. We also support these type of events, for instance, JOBIM. I talked about these emerging scientific events, and also visits if you're interested. Of course, as was mentioned by Morgan, we need people to help us make things happen. So if you have ideas, if you want to join, even for a very small task, just come to me. And that's what I want to finish with. If you have any questions now, maybe the time is limited, but after, I will take the boat trip. So if you're on the boat trip and you have questions, you can reach me, and I will be there, of course, for the rest of the conference. Thank you. No questions. Everything's crystal clear.
Hello, I wanted to ask, when you mentioned about helping you talk about grant for a project, in sort of some ideas, like you, okay.
Yes, yes.
So
small grants. CNRS doesn't provide us so much money, but this would be a small grant, yes.
Okay, so has to be affiliated with the group? I mean, is it like individual grant, or it's a project specific, or specific to the group we are affiliated with?
So I don't know what you're talking about. I guess you're talking about this, what I called in French, atelier exploratoire, if you have an emerging scientific topic. So there are conditions which I didn't take time to list there, but maybe we can discuss that after. But one of the conditions is that it has to involve at least 2 different places in France. It's only French, because that's what the CNRS asked us. It has to involve at least 2 research facilities, and it has to be interdisciplinary. It cannot be only one discipline.
Thank you for the presentation and the update about JOBIM. You showed the domains on which you have activities, some of which are recurrent, some of which are up to date with the AI. But and we all know all these challenges for bioinformaticians. Most of them, we see them at IFB on the point of view of infrastructure, service delivering, skills, skill delivering, training, and so on. But I wanted to ask you, for you, what are the main scientific challenges that are in front of us for the bioinformatics communities? What are the topics where new science will have to be done, and we are on it, but still need to go one step ahead?
This is a very tricky question. I don't know if I have to thank you for that question, because end of the afternoon, out of the blue, like this. Okay, I cannot really answer for the whole community. This is something we should, so I have an easy answer, which says normally next year we should gather and discuss about that. What comes to mind for me is things which are related to metagenomics and maybe metaproteomics. Okay, like the big picture, you know, the more data, more complicated data, etc. So I don't know if that answers correctly your question, but and of course AI, but we don't know exactly what to do with AI in terms of methodology. So
I don't have the answer either. I'm just wondering, in science, what are our challenges as a community? I'm not sure it's obvious, but I think it's something that JDR-BIM, since basically it's around science, I think it would be nice to have a sort of brainstorming about that.
That's what we try to do, but as usual, we lack time to sort of take time to think about it. But that gives me the opportunity to say something. So we have, at the moment, we have 6 working groups, but some could in the future disappear if it's out of date, and some other could appear. And so that's also something that you can let me know if you have new ideas, and if you think there is a community which is not represented there, then that's the occasion to discuss. So Stéphane is from Nantes, so maybe we can discuss in Nantes.
Yes, but I think it is important to say it here. If you go back to the committees and the presentation that was triggered just before, one thing about the future is thinking about what can be the misuse of the bioinformatics. It was already mentioned about the European health data, but I see more and more demands or remarks about the fact that our methods can be changed to do something weird. And this is just to give that to everyone, and also we can discuss, we can continue to discuss, but this is an emerging topic that I have discussed with many companies or partners.
Okay, thank you.
Hello everyone, so we are going to present JBIF in French, but if anyone has some question after, you can come in the main hall and we will answer the question in our stand. Donc bonjour à tous, je suis Elisabeth Elec et je suis la présidente de JBIF, donc l'association des jeunes bioinformaticiens et bioinformaticiennes de France, et je suis accompagnée de mon trésorier, donc Jérémy Rousseau, qui est doctorant au MNHN à Paris. Et donc aujourd'hui, on va vous présenter un peu JBIF. Peut-être que vous en avez entendu parler autour de vous durant ces dernières années. Donc l'association des jeunes bioinfos de France. Donc là, vous avez un peu toutes nos petites têtes du bureau et du CA. Donc vous allez pouvoir nous retrouver tout au long de la conférence. On est plusieurs sur place. Maintenant, on commence à avoir une communauté qui a bien grossi ces dernières années. On est à environ 80 adhérents tous les ans. On a une quinzaine de bénévoles qui sont actifs et qui nous aident sur nos différents projets qu'on va vous présenter juste après. Et à côté de ça, on a une grosse communauté sur Discord. Donc on vous parlera aussi de ça un peu après, où on va partager les dernières actualités en bioinfo, on va discuter entre nous des jobs, etc., des emplois. Et nous sommes présents aussi sur les plateformes YouTube et Twitch, et on va vous présenter pourquoi juste après. Depuis l'année dernière, on fonctionne sous la forme de groupes de travail. Donc on en a un certain nombre qu'on a mis en place. On a des groupes de travail autour des différents événements qu'on va pouvoir organiser tout au long de l'année. On a un groupe de travail JBIF à JOBIM, où on va mettre en place le workshop qui a lieu juste avant JOBIM, mais aussi notre stand, etc. On a des groupes de travail autour de la fête de la science, car ça nous tient à cœur de vulgariser la bioinformatique, parce qu'on sait que c'est pas très facile de vulgariser nos sujets qui peuvent être des fois très compliqués. Donc en tout cas, on essaye de le faire au cours de la fête de la science. On a un GT BioStream qu'on vous présentera juste après, et 2 GT sites web et statuts qui nous ont pris aussi pas mal de temps l'année dernière, mais qu'on ne va pas détailler ici. En gros, vous voyez, rejoignez-nous. On est à la recherche de bénévoles sur nos différents projets. Il y en a beaucoup. Donc si vous êtes jeune bioinfo, peu importe le statut, étudiant en master, doctorant, postdoc, ingénieur ou en recherche d'emploi également, n'hésitez pas à vous joindre à nous. C'est un super moyen de rencontrer du monde et puis de s'impliquer pour la communauté. Et je vais laisser la main à Jérémy.
Donc JBIF, c'est tout d'abord la fête de la science qu'on réalise à la Cité des sciences et de l'industrie à Paris tous les ans, au début octobre. Donc c'est le moment où on va faire de la médiation scientifique, vulgariser la bioinformatique au travers d'ateliers qu'on est en train de créer et qu'on crée. Donc chaque année, on se calque sur la thématique de la fête de la science. Cette année, ça va être la saveur savante, donc tout ce qui est autour du goût, des saveurs, et on va y intégrer donc la bioinformatique, ce qui va être assez compliqué. Donc pour réaliser tous ces ateliers, on est toujours à la recherche de bénévoles. Donc si vous êtes intéressés, n'hésitez pas à vous rapprocher de nous. Mais on peut aussi vous présenter sur notre stand les ateliers des années précédentes, notamment 2024 et 2025. Donc c'était l'océan en 2024 et les virus en 2025. Et JBIF, c'est aussi avant tout son workshop annuel qui a eu lieu juste avant JOBIM, lundi, où on aborde à ce moment-là des thématiques diverses et variées. Ça va de la reproductibilité en passant à la médiation scientifique et cette année à l'employabilité. Par exemple, si je prends le cas de cette année, on a pu organiser des tables rondes pour présenter les réseaux, mais aussi une table ronde avec des acteurs professionnels de la bioinformatique, qui soient du secteur public ou privé. Donc des personnes qui sont spécialistes, qui sont activement présents dans les laboratoires ou dans les équipes et qui recrutent souvent. Et donc le workshop cette année a pu être réalisé grâce au soutien de l'ISCB Student Council, mais aussi de la SFBI et également du comité d'organisation des JOBIM.
Je passe à un autre gros projet de JBIF. Peut-être que vous en avez entendu parler ces 2 dernières années. On a mis en place les biostreams. Donc c'est des lives qui vont tourner autour de la bioinformatique, peu importe les sujets. Et donc on se donne rendez-vous en général chaque premier mercredi du mois, où on anime ces lives avec Benjamin qui est ici présent et aussi Magis qui est notre régisseuse. Et on va discuter de plein de choses. Donc on a commencé par présenter justement tous les réseaux que vous avez pu voir aujourd'hui. Mais on a aussi, pour cette année, commencé à discuter un peu plus projets scientifiques, thématiques scientifiques, avec notamment une émission autour de la bioinfo marine et également des biostreams sur les maladies rares et comment on va poser des diagnostics pour ces maladies rares. Donc actuellement, les lives ils sont accessibles en rediffusion. Vous pouvez les regarder sur notre chaîne YouTube de JBIF. Il y en a 13. Et on tient aussi au passage à remercier tous nos intervenants et intervenantes qui sont venus discuter avec nous sur le biostream, dont on a pu se croiser pour certains. Merci beaucoup parce que ça fait vivre aussi la communauté. Et puis on sait que c'est un format qui n'est pas facile, mais en tout cas vous jouez tous le jeu, donc merci. Et si vous avez des idées de biostream, n'hésitez pas à nous contacter. Je continue avec du coup notre dernière slide. On a fait assez court pour cette année. JBIF, on est présent dans toute la France. On ne fait pas que les projets qu'on vient juste de vous présenter. On est aussi actif sur les bioinformations, la newsletter de bioinformatique. On organise des JBIF Pub dans toute la France. Donc, ce sont des after-work informels qu'on essaye de mettre en place plusieurs fois par mois si on le peut. Donc, on est sur pas loin de 17 villes où il y a déjà eu des JBIF Pub. Donc, si vous voulez en organiser, vous pouvez aussi prendre contact avec nous. Là, vous avez un énorme QR code sur la diapo. Donc, si vous voulez joindre le Discord, on est 667 membres actuellement. Notre objectif, c'est toujours plus. On aimerait bien que les jeunes bioinfos, mais pas que, se rejoignent à nous sur ce Discord. Et puis, en quelques mots-clés, JBIF, c'est tout ça. C'est des afterworks, c'est des rencontres, des tables rondes, de l'entraide, les biostreams, du réseau aussi. On est présent sur LinkedIn également. Donc, si vous voulez regarder un peu ce qu'on fait, vous pouvez regarder notre page. De la médiation, de la vulgarisation. Cette année, une fois de plus, on a aussi co-organisé les job fairs avec la SFBI. Donc ça a plutôt bien marché et on est très content qu'il y ait eu autant de participants cette année. Petit point également à noter, si vous êtes des potentiels recruteurs, n'hésitez pas à vous positionner l'année prochaine parce qu'il nous en manquait quelques-uns. Donc voilà, JBIF en quelques mots, c'est tout ça. Et on tenait également à remercier notre marraine qui est ici présente, Morgane Thomas-Chollier. Merci beaucoup d'avoir accepté une fois de plus d'être notre marraine. Et merci à tous. Je voulais rajouter un mot ? C'est bon. Merci.
Je vais parler aussi sur la dernière.
Ah, pardon. Désolée.
Merci beaucoup pour cette présentation. Alors, peut-être que je n'ai pas très bien écouté, mais il me semble que vous avez oublié de préciser que JBIF fait partie d'un réseau international aussi, le réseau des RSG pour Regional Student Council, pour Regional Student Group. Et donc, vous faites sûrement la branche la plus active, en fait, à l'international. Et j'aimerais savoir, est-ce que vous avez peut-être prévu dans les prochains temps un événement justement avec d'autres groupes dans le monde ou en tout cas en Europe?
Si tu as la réponse.
Merci Victor, bien sûr. Alors effectivement, on a oublié de le préciser parce que là, on voulait plutôt acter les différents projets qu'on met en place actuellement. Mais effectivement, on est la branche française du coup et on est rattaché au Student Council. Actuellement, on a un projet, enfin en tout cas, moi je suis rattachée à la conférence européenne, donc c'est l'European. Tu peux me retirer si tu as le micro peut-être? Parce que l'acronyme il est un peu long.
Oui, alors on travaille, Elisabeth et moi, sur l'European Student Council Symposium qui aura lieu durant le CCB en septembre. Donc si vous souhaitez venir, n'hésitez pas, ce sera super sympa. Et le CCB, c'est l'équivalent de JOBIM, mais version Europe. Voilà.
C'est ça. Et pour répondre à ta question sur les projets futurs, on aimerait bien se rapprocher de notre équivalent JBIF en Belgique, mais aussi au Luxembourg, qu'on avait commencé à discuter pour mettre quelque chose en place, notamment au niveau de notre workshop, donc juste avant JOBIM. Ça aurait été l'occasion parce qu'on était à côté, on était à Strasbourg, mais le temps nous a manqué. Mais effectivement, on aimerait bien, maintenant que tous nos projets commencent à être bien mis en place et qu'on a pu mettre un certain nombre de choses où maintenant on se débrouille bien et on sait comment ça tourne, effectivement, c'est nos projets, au moins pour l'année prochaine, de diversifier un peu plus le réseau.
Merci pour la présentation. Pour tout ce qui est organisation d'événements, on va dire plutôt informels du genre JBIF Pub, c'est plutôt d'une initiative de... Enfin, JBIF Pub ou autre, mais c'est surtout à l'initiative de groupes d'étudiants ou de gens qui sont déjà membres et qui disent je vais organiser un JBIF Pub cette semaine.
De toute personne qui veut en organiser un, il nous contacte. C'est à vous de trouver le bar et nous, on peut se charger ensuite derrière de tout ce qui va être communication, diffusion sur les réseaux pour médiatiser tout ça. Et on se charge aussi, par exemple, de tout ce qui va être lien d'inscription pour permettre à tout le monde de s'inscrire.
Je peux rajouter un mot ? Il y a aussi pas mal de ce genre d'événements informels, un peu façon JBIF Pub, qui sont mis en place par des groupes d'étudiants, effectivement. C'est vrai que ça bouge plus facilement quand on va avoir des masters dans les villes qui sont déjà mis en place. Mais il y a quand même des villes où il n'y a pas forcément des personnes qui ont un gros groupe ou un gros réseau comme rattaché à un master. qui arrivent à mettre en place ce genre d'événement. Vraiment, il suffit de nous envoyer un message et nous, on a déjà en tête quelques personnes avec qui vous pourriez vous mettre en contact justement pour faire ce type d'événement. Donc en tout cas, il ne faut pas hésiter de nous contacter. Et après, nous, on vous aide complètement pour la logistique, etc. De même, là, on parlait des JBIF Pub, mais ça peut être une table ronde. Et c'est vrai qu'on n'en a pas parlé, mais ça nous est arrivé d'intervenir au Mix Day de Montpellier. On est aussi déjà passé par le Big Day, donc les journées de conférence du Master de Bioinfo de Rennes. Donc voilà, il suffit de nous contacter. Nous, on fait au mieux et si on peut se déplacer, on passe aussi dans les formations ou dans les événements.
Super, merci.
Merci. J'avais une petite question sur vos biostreams. En fait, quel est le public que vous visez
On vise à peu près tout le monde, tous ceux qui sont intéressés. L'objectif, c'est que ce soit le plus ouvert, le plus accessible à tout le monde. C'est vraiment
un background un peu science quand même.
Oui, un petit peu de background science, mais vraiment, ça reste très, très accessible.
Oui,
toute personne intéressée peut le suivre.
N'importe qui d'une autre discipline.
Okay. Et l'objectif, c'est vraiment le fait que ce soit un stream. Ça permet aussi de l'interaction. Donc, s'il y a des questions, s'il y a besoin de reformuler, de reposer, c'est là où ça devient intéressant.
Merci beaucoup.
Je rajoute aussi un petit mot pour les biostreams. En général, on fait après coup, puisqu'il y a la rediffusion. On est à peu près sur 200 personnes, 200 vues, voire plus pour certains streams. Donc ça touche quand même essentiellement la communauté Bioinfo. Mais il y a certains streams peut-être plus thématiques scientifiques qui étaient plus accessibles. Je pense au live sur la biomarine, où il y a eu pas mal de gens qui ont suivi, mais plutôt biologistes. Après, c'est sûr, les réseaux Bioinfo, ça concernait quand même plutôt la communauté ici présente. Donc ça dépend vraiment en fait des streams qu'on fait. On a plein de questions.
C'est bien. C'est une question un petit peu dingo. J'essaie d'encourager certains pays européens, en partie aussi déjà, qui n'ont pas beaucoup de visibilité et qui travaillent pas mal en bioinformatique, à créer justement des sociétés pour essayer de gagner cette visibilité. Sauf que les vieux, j'arrive pas à les convaincre. Vous seriez partant pour aider, et sachant que dans ces pays-là, les jeunes parfois sont pas mal isolés. Vous serez partant si je vous donne un ou deux petits contacts pour faire des trucs?
Carrément.
Ok, c'est le but de notre association aussi.
Si on peut aider à développer des réseaux comme ça, ça nous va.
Ça fait partie de nos missions de rassembler les jeunes bioinformaticiens de France en l'occurrence, mais on essaie aussi de se rassembler à l'international, comme disait Victor tout à l'heure. C'est un de nos projets. Donc effectivement, oui.
Ok, c'est noté.
Juste deux mots, un grand merci du coup à l'ensemble des réseaux qui ont présenté, donc le réseau Mérite tout à l'heure, l'IFB, le JDR-BIM et puis JBIF. Un grand merci à vous.